the bicentenary birthday of Gregor Johann Mendel. I am delightful to begin today's session, that is blooming the birth of new science, by inviting our honorable chairman of this session, Dr. Akhilesh Tyagi, and co-chairman, Dr. A.K. Shishani, to grace the dais. I would also like to invite the speakers of this session, Dr. T. Balram, Dr. P.K. Gupta, and Dr. Vishwanathan C. Now, I would like to welcome the conveners of this session, Dr. Vishwanathan C. and Dr. Gyan Prakash Mishra. And also, I welcome Dr. Prachi Yadav, Dr. Hari Krishna, and Suman Datta, rapporteurs of this session. May I now request Dr. Vishwanath C. on the podium to introduce the chairman and co-chairman of this session. May I now request Dr. A.K. Singh, Director, to welcome the chairman and co-chairman and also speakers with a bouquet of flowers. May I now request Dr. Vishwanath C. on the podium to introduce the chairman and co-chairman of this session.
Good morning to all of you. Thank you for giving me the pleasant duty of introducing the chair and co-chair in this important session. I welcome Professor Akhilesh Tyagi who has kindly agreed to chair this session. Professor Tyagi, born in 1956 at Bulandshahr, India, and he has obtained his PhD from Delhi University, and then he has carried out his postdoctoral research work at Germany in two different universities, Dusseldorf University, Germany, and Munich University, Germany. Then after that, he has joined at University of Delhi as a reader, and then from that position he has rose to the professor position, and then later he moved to National Institute of Plant Genome Research as director, and then he has also served as executive director of NOBI. And then during this 36 years of his research career, he has mentored over 120 postdoctoral fellows, doctoral students, and master students. He has made significant contribution in the functional genomics as well as in the area of genomics, and everyone remember very easily he has his contribution in the genome sequencing of rice, tomato, and chickpea. He has over 3,000 research publications in the high impact journal, and he has decorated the national academies, such as he has served as Secretary General, National Academy of Sciences, and he has served as Vice President, Indian National Academy, and he has also served as President, National Academy of Sciences. Because of his vast contribution in the research and nation building, he has been awarded with several honors and awards. Some of them include National Bioscience Award as early as 1999, and then San Francisco World Technology Award in 2003, and International Year of Rice Research Accomplishment Award in 2004. And there are a long list of awards he has been received, and he is Fellow of National Academy of Agriculture Sciences, New Delhi, Fellow of Indian National Academy of Sciences, and National Academy of Sciences, India, and Indian Academy of Sciences. He is also a Fellow of Third World Academy of Sciences. So I welcome you, sir, with this brief introduction. I also welcome the co-chair of today's session, Dr. Ajit Kumar Sashani. He is again born in 1967 at Sambalpur, Odisha, and he is an alumni of the Indian Agriculture Research Institute. He has completed a PhD from IARI in 1996, and then he has done his postdoctoral research at Purdue University. In fact, he joined ICR as a scientist to begin with, and then he moved to the CSIR CMAP, where he has carried out extensive work on medicinal plants. And to his credit, he has over 250 publications and more than 100 patents for various metabolite of importance that he has identified from the medicinal plants. He has been awarded with several awards and honors. This include Indian Drug Manufacturer Association Award for the Best Patent Team Award, and SAM Award for Essential Oil Association of India, and CIM Fellow Award of 2007, and various award he has received, including the CSIR Technology Award for Biological Sciences in 2012, and he has been elected Fellow of National Academy of Agriculture Sciences and NASI, and he is also a fellow of various other professional societies of the country. So I welcome you, sir, to co-chair this session. Thank you, Ramesh. Now I request Chairman to carry forward.
Honorable and distinguished luminaries of the area of genetics and related sciences, it's a great pleasure to be here on this occasion when we are celebrating 200 years of the birth of great Gregor Mendel. I am sure you enjoyed the session yesterday, and today we have a very distinguished panel of speakers in this session, which is named as Blooming Birth of a New Science. And these speakers are, with slight change, Professor P. Balaram, Professor P.K. Gupta, and Professor C. Vishwanathan. I see that Professor Painter could not come due to some reasons. Now, coming to the first speaker, Professor Padmanabhan Balaram has been working on the molecular biophysics unit of the Indian Institute of Science, Bangalore. He has been director of the institute and has also been awarded Padma Bhushan by the government of India for his very distinguished work. The work which he has been doing is very difficult for me to define, but I understand that he has been working on structure, conformation, and biological activity of design and natural peptides. And this particular area he has nurtured and has produced a large number of students trained in this particular art of science. Of course, we also know him as the chief editor of Current Science, and like me, many of us have enjoyed his editorials on different themes related to science as well as society. And this time he is going to talk to us about Mendelian genetics and Darwinian evolution, bridged by the chemistry of DNA. Obviously, these are two great works of Mendel and Darwin, which have changed our thinking about biology altogether. And bringing them together are certain bridges which people are trying to understand for a very long time and are still busy to decipher the intricacies of these two founding principles of biology. So without standing for long between you and Professor Balaram, I would like to invite Professor Balaram to deliver his lecture. Professor Balaram.
Good morning. Thank you very much, Professor Tyagi, for that introduction. And I must confess that when Dr. Singh and Dr. Vinod invited me to come to this symposium, my first confession was that I'm not a geneticist at all. And therefore, I ought not to be speaking on a symposium honoring Mendel on his 200th birth anniversary. I was, however, persuaded to come, and therefore I have prepared fairly hard for this lecture. And my very first slide shows you two of the conceptual pillars of biology. Darwin, who gave us the ideas of natural selection and evolution, and Mendel, of course, who gave us our ideas of heredity, the transmission of characteristics through generations, and so on. I'm going to try and bridge them with a subject that I know to some extent, and that is chemistry. And I thought if I'm going to bridge two of the conceptual pillars of biology, I must put in the center a bridging piece. And that bridging piece is really the book by Erwin Schrödinger, the physicist who set off the revolution in quantum physics in the early part of the 20th century. And Schrödinger reflected in a series of famous lectures, which I will come to in 1944, on the problems of biology. I went back to the literature and asked myself the question, and I never studied biology. So I'm not trained formally in biology. Everything that I know by biology is what I have learned later on. But one thing that students who are not students of biology must realize is that biology is a subject that is best learned as you age. You understand it very much better. And one of the things about the process of aging and life is it's teaching you about biology almost every day. We talk about Mendel and we talk about genetics. Mendel, of course, doesn't use the term genetics. So where did the term genetics and genes originate? This, of course, originated in a book which appeared in German, which I picture on the other side, which is called, translated, The Elements of the Exact Theory of Heredity by the Danish scientist Wilhelm Johann. And what Johann really did was he defined some terms. And I want you to read something from the slide. He says, for example, the science of genetics is in a transition period becoming an exact science, just as chemistry in the times of Lavoisier, who made the balance an indispensable implement in chemical research. Quantitative chemistry's origins really are in the 18th century when Lavoisier invented the balance. It turns out that much later, even the origins of biochemistry are with the balance where you try to make sure that the carbon that you burnt is matched on the other side by the carbon dioxide that is produced by living organisms. He also says something interesting. You will note that at the top of this slide, I have the title of the very first lecture in your symposium program. And that very first lecture was a sort of transliteration into English of the Hindi title. If it had been written in Hindi, I would have been able to put it on the slide much more easily and then get a translation from someone. So I called up one of my students and asked him, can you translate this for me? Because I don't understand the first word here. And then he said, that is the Hindi word for genetics. Now, of course, you can go back into the origins of language and ask the question, did people in Indian languages worry about genetics? Here it is that Johansson says so wonderfully. He says here, it's a well-established fact that language is not only our servant when we wish to express or even to conceive our thoughts, but it may also be our master, overpowering us by the means of the notions attached to current words. So Johansson's very aware of the fact that language is very important. You know, after the Second World War, when after the war was over, many assessments of Winston Churchill came to the conclusion that what Churchill did during the war was to weaponize the English language and make the people stand up to all the difficulties. Here, Johansson says, I proposed the terms gene and genotypes and some further terms as phenotype and biotype to be used in the science of genetics. This is an English lecture that he gave, which has appeared in the American Naturalist in 1911. And when I read this, I found that if you read the very old papers, you don't have to have gone to a course in genetics. You can actually understand what they're saying because much of it is very simply said. Towards the end of his lecture, Johansson also says that he must emphasize this book, which appeared in 1901. And this book, of course, introduced the idea of mutations. These dates are very, very important. Johansson's work begins in 1909 or thereabouts. De Vries' work is 1901. And these are very important. These are very important in the history of science because science was changing at this time. Remember that the 19th century was a time when physics, chemistry, biology did not have the sharp divisions as they have today. Everybody really in the 18th and 19th centuries was a natural philosopher. We all get degrees, doctors of philosophy, but we are anything but philosophers. And the origin of the degree itself comes from the earlier parts of the 18th and 19th centuries. Here he talks about mutation. He says, like the head of Janus, it looks at once forward and backward, trying to reconcile at least partly the antagonistic ideas of continuity and discontinuity in evolution and heredity. This idea of continuous change and discontinuous change was really the perceived conflict between Darwin and Mendel in later years. There was no conflict between Darwin and Mendel. They hardly overlapped, although Darwin was a bit senior, Mendel came a little bit later. Darwin was largely unaware of Mendel's work. Mendel was aware of Darwin's work, and contemporary historians have gone back to available records, letters, and so on to analyze it. He also concludes this at the end by saying, these qualities have made the work of De Vries too eclectic for the stringent analytical techniques of modern genetics, a tendency which has in recent years found a true home in American science. Remember that American science was just beginning to move, German science was beginning its decline, which will accelerate during the first decades of the 20th century. Can you just move the slide forward? It's not moving from it. All of you know, and I'm sure it's discussed in this conference, Fisher's famous paper in 1936, where he raised this question of whether Mendel's results were really true or whether Mendel's results were made to fit a theory which appeared to be extraordinarily satisfactory to explain many observations. Fisher, of course, was one of the fathers of modern statistics. But Fisher's work has been questioned in later years. In 1984, in the Journal of Heredity, there is this paper which I think you should read. But what it says here in this paper is that if Fisher's papers had been ignored, as Mendel's was, this rebuttal would be unnecessary. Unfortunately, the rebuttal later on has come from many sources, and there are papers in statistical science which try to reconcile the Mendel-Fisher controversy, but I will not get into that. Instead, I'm going to come to the middle of the 20th century. We are moving fast forward now. We've started in the 1850s, 1860s. We have moved through the 19th century, and we are coming to 1944. In 1944, after having fled from Hitler's Germany, Erwin Schrödinger, who gave us the Schrödinger equation, one of the starting points of quantum mechanics, gave a series of lectures in the Institute of Advanced Study in Dublin. These lectures have been celebrated in later years for various anniversaries, and many years later, the 100th anniversary of these lectures will be celebrated undoubtedly in 2044. But the Dublin lectures are very famous. Here was a physicist now asking a question, what is life? And when Schrödinger asked this question, what is life? He had in fact gone back to the literature and read all the early work in biology. And effectively, in asking what is life, he was asking what is the unit of heredity? And he asks, what is the gene? Now, in the anniversaries that have followed, a Nature editorial in 2018 said this. Here it says, here Schrödinger made the auspicious proposal that the genetic material is an aperiodic crystal, a structure with a specific but not periodic arrangement of atoms that somehow guides the development of an organism. That vision resonated with Crick and Watson as they contemplated the structure of DNA. The editorial concludes by saying his timing was perfect to raise this question in public and write a book. Schrödinger presented the problem of life as a puzzle, posed to no single discipline. And his timing was perfect. Biology was already changing from a largely descriptive, historical, and organismal science to a mechanistic and microscopic one. We by now knew about chromosomes. We knew these structures inside the cell. Cytology was already there. And the microscopes were advancing very fast, and cell biology was on the horizon. Schrödinger, of course, had a deplorable personal life. But if we set his personal life aside and only look at the book that he wrote, the book was extraordinarily influential. There are a few books in science which are extremely influential to a generation of scientists. There's a book by Paul de Criff called The Microbe Hunters, which inspired a generation of biochemists and microbiologists. Schrödinger's What is Life inspired a generation of physicists and chemists to enter biology. Schrödinger's book in 1944, Schrödinger's book, in case you want to buy a copy today, it's still worth reading because some of the questions are posed with the clarity that only a physicist looking at the fundamental structure of matter and the structure of atoms can simplify. Very important in science. It's very important in science to simplify your problem, to articulate what your question is before you can actually answer it. Today, in the modern world of biology, we are more driven by technology and by techniques than by the conceptual foundations of biology. Schrödinger begins his book with a quotation from Spinoza, translated, which reads, "There is nothing over which a free man ponders less than death. His wisdom is to meditate not on death, but on life." And this is something that all of us carry through to the very end. It's better as you age not to worry about death, but to really worry about biology, because biology is life. And then he says, he talks about the gene. What we locate in the chromosome is the seat of this difference. We call it in the technical language a locus, or if we think of a hypothetical material structure, we underlying it, it's a gene. This is Johansson's gene now. The question then in 1944, before a large audience which Schrödinger articulated, what is the gene? Ironically, as always happens in science, it was exactly at the time that Schrödinger was giving his lectures that Avery published his famous work on DNA as the pneumococcal transforming principle. And I will come to that in a moment. Here is Schrödinger's question. Schrödinger asked, how can the events in space and time, which take place within the spatial boundary of a living organism, be accounted for by physics and chemistry? He responded, the obvious inability of present-day physics and chemistry to account for such events is no reason at all for doubting that they may be accounted for by those sciences. And in the ensuing 70, 80 years since Schrödinger wrote these words, they have in fact been accounted by the laws of physics and chemistry. We now know that there are new unknowable laws which still exist for physicists to learn in biology. In fact, today, what physicists actually ask, there's a famous quote which says, "Ask not what physics can do for biology, ask what biology can do for physics." This is paraphrasing something that John F. Kennedy said in his famous inaugural speech in 1960 when he was sworn in as the American president. He asked, "Ask not what the country can do for you, but ask what you can do for the country." Schrödinger on Mendel. Schrödinger has a paragraph on Mendel. He describes Mendel's work, how Mendel's work was resurrected in the early part of the 20th century. But he says his paper was forgotten and was only rediscovered in 1900. And then he gives the names of the scientists who rediscovered Mendel's paper in 1900. But 1900 is a very important year. 1900 was the year in which Planck published the idea of the quantum. So if you go back and ask, what was the quantum revolution, the starting point? It's the year 1900. The word quantum is as important to physics as the gene is to biology. All of these ideas were coming at about the same time at the beginning of the 20th century. So you can see when we look back, the 20th century will become the defining century in which 19th century biology, physics, and chemistry are completely integrated with 21st century technologies which are taking us forward. Schrödinger, for example, because of his work in statistical physics, is worried about the gene. If the gene is like a drop of liquid, then of course a drop of liquid, everything is disordered. Entropy increases. Statistical physics has its origins in the work of Ludwig Boltzmann in the 19th century again, more or less contemporary with Darwin and Mendel. Boltzmann thought about heat. He asked, what is heat? And then came to the idea that heat is a motion of atoms. And the motion of atoms led him to the idea of entropy. And therefore you can see that atoms, entropy, these are the driving concepts for Schrödinger to worry about biology. They are important. Even today, biology does not violate the laws of thermodynamics. It does not violate any fundamental physical law, but biology is taken as a whole with its surroundings. Today, when biologists talk about sustainability, biologists talk about ecology, they are talking about the environment as a whole, the exchange of energy, the exchange of information, the exchange of material between any organism and its environment. And then he says that the gene is probably a large protein molecule. At that time, proteins were beginning to occupy center stage in biochemistry. And this was also the development of biochemistry. Remember that if you take the 20th century, in the words of Arthur Kornberg, the first 20 years or so of the 20th century were the age of the microbe hunters. Everything comes from medicine because human beings are actually sometimes far more interested in medicine than they are in agriculture. Both of these are two arms of biology. In thinking about the 20th century, remember the first 2 decades are the century of the microbe hunters. But after the 1920s to the 1940s, biochemistry is driven by nutrition. The vitamin hunters who hunted for the diseases which are caused by nutritional deficiencies found all the vitamins. But the biological role of the vitamins was not known. That they function as coenzymes became clear only between the 1940s and the 1960s. And that, of course, might be the 2 decades of enzymes and proteins. So everybody at that time was beginning to be interested in proteins. It was also thought that genes were proteins. He in fact says that these atoms are all held together that will give the gene a certain stability. because the gene now has withstood centuries. It's been transmitted through the generations. And here is where Schrödinger says famously the words aperiodic crystal. He says physicists in the early part of the 20th century were dominated by crystal physics. Remember that Bragg's law appeared in 1911 or 12. Laue's demonstration of X-ray diffraction by crystals was a little bit earlier than that. Röntgen's discovery of X-rays was at the end of the 19th century. And therefore, physics was using X-rays and studying crystals. Crystals and space groups, regularity and symmetry dominated physics. Optics was dominated by crystal optics. And here you can see that the most essential part of a living cell, the chromosome fiber, may suitably be called an aperiodic crystal in physics, which has hitherto dealt only with periodic crystals. And he talks about the difference. The difference in structure is of the same kind as that between an ordinary wallpaper in which the same pattern is repeated again and again, and regular periodicity, and a masterpiece of modern embroidery, say a Raphael tapestry, which shows no dull repetition, but an elaborate, coherent, meaningful design traced by the great master. And now, of course, I've pictured this on my slide with a wallpaper with symmetry and a Raphael tapestry there, which is his famous painting of the angels. And then he says, organic chemistry, and this of course gladdens my heart because this is the subject which I learned in my master's program. Indeed, in investigating more and more complicated molecules has come very much nearer to the aperiodic crystal, which in my opinion is the material carrier of life. And therefore it is small wonder that the organic chemist has already made large and important contributions to the problem of life, whereas the physicist has made next to none. Schrödinger said this in 1944. The structures of proteins and the nucleic acids were just on the horizon. 1953 was the Watson-Crick double helix. 1951 was Linus Pauling's alpha helix in proteins. 1954 was G.N. Ramachandran's triple helical structure of collagen. The hereditary mechanism is dealt with in Schrödinger's lectures, and you can see the kind of pictures that he used. Biology grew with drawings, hand drawings, and you can see some which have been used here. They may be different. Schrödinger frequently quotes the German poet von Goethe, and here is a quotation translated into English. Being is eternal, for laws there are to conserve the treasures of life on which the universe draws for beauty. So it turns out that the universe has really drawn for its beauty all the beauty that you see in biology by faithfully transmitting information over generations and at the same time varying that information. He talks about mutations, and this is where you see the conflict at that time. Is evolution a discontinuous process, or is evolution a continuous process? Today we have reconciled this because we understand the basis of mutation. The chairman of the session has sequenced the rice genome. You understand it in all its detail, and therefore you are not surprised by mutations. But at that time people were. So when you see a jump for a physicist, it immediately attracted Schrödinger. He says maybe the jumps are like the jumps that occur in quantum theory. And this is true. In quantum theory, there are two energy levels. There's no intermediate states between those. That's what makes quantum theory so difficult to reconcile with everything that we see in real life where there is continuity. Similarly with a mutation, there's nothing in between. A mutation happens, the gene has transformed. There is no intermediate state. This is unlike a chemical reaction, for example, where something really changes and there is intermediate. He says here that De Vries' mutation theory is figuratively the quantum theory of biology. And then he says, but quantum theory was but 2 years old when De Vries first published his discovery in 1902. Small wonder that it took another generation to discover the intimate connection. It is worth looking back at history, especially if you're students. If you don't know how things have come about, it's very hard to even visualize what might happen in the future. It is even hard to articulate the scientific challenges of the future. Sometimes we confuse the technological challenges which are immediate to solve real-life societal and social problems with the problems of science. Schrodinger's questions sometimes are interesting. He says the gene has been kept at a temperature around 98 degrees Fahrenheit during all that time. How are we to understand that it has remained unperturbed by the disordering tendency of heat motion for centuries? So he is worried about thermal order. And this is true, because in molecules, you're always worried about thermal order. It's chemistry, the bonds between atoms, which allow molecules to resist the disordering tendencies of thermal motion. But we also know that beyond a certain temperature, there is no life. But he says it is of limited value to understand all of this, as the enigmatic biological stability is traced back only to an equally enigmatic chemical stability. Here I'm going to digress a little bit, because we are in 1944. Avery, I will come to him, published his paper in 1943. But it turns out that if many of you believe that Mendel was ignored, Mendel was not really ignored. The circumstances in the 19th century did not permit Mendel's work to be adequately understood. If you want to find a scientist who has been ignored, Avery made probably the most important scientific discovery of the 20th century, namely the discovery of DNA as the genetic material. Nevertheless, Avery was ignored, and all of Avery's recognition has really largely come after Avery passed away. But I'm going to show you a paper. This is a paper that I like very much. This is a paper written by Linus Pauling, and the paper appeared in 1949. The year is important for two reasons. One, of course, is it comes between the Schrödinger Dublin lectures and the Avery publication and the Watson-Crick publication of the structure of DNA. 1949 is the year that I was born. So when I see a paper that was published in 1949, it immediately tells me something about time. We all remember the time of our birth, and we never forget it. We celebrate it also. And the reason for this is, of course, we should celebrate something in science that happened around the same time, and this is the Pauling paper. I'm going to tell you a little bit of history, little bit of protein chemistry, not very much. Pauling wanted to study hemoglobin. The reason he wanted to study hemoglobin is sort of interesting. He was working on minerals which contain iron. His interest was to measure the magnetic susceptibility of iron, and there was very little money in those days. These were the years of the Great Depression, the years that preceded the Second World War. So when there was no money, and in India sometimes we have worked in situations where money has been scarce, one sort of sympathizes with people who work under those conditions. Now, Pauling was ingenious. The Rockefeller Foundation for Medical Research had just been set up, and they wanted to fund what Warren Weaver, the first director of the Rockefeller Foundation, called the new and developing science of molecular biology. Weaver did not talk about molecular biology the way all of you talk about it. Weaver actually was thinking about the connections between the kind of work that Pauling was doing and the kind of work which was a very much more structural approach to biology. And one of the very first programs that Weaver actually funded was Pauling's work. When Pauling realized that he couldn't get money for studying iron minerals, he decided that he would study blood, because hemoglobin has iron right in the middle, and therefore he could now get money to study the magnetic susceptibility of hemoglobin. He could use the same balance to study the magnetic susceptibility of iron minerals. Pauling, of course, was a remarkable man. He began his career in quantum mechanics, directly going to work with Schrödinger's disciples, Heitler in London, who worked on the hydrogen atom. So Pauling was, in fact, a polymath whose work spanned all disciplines from physics to chemistry to biology. And then, of course, he says, you would have read all of this, hemoglobin is easy material to get. And as he says, all you have to do is catch someone who's smaller than you and prick their finger, you'll get hemoglobin, drop of blood. And then if you get the red cells into a test tube and add a little bit of distilled water, the red cells will undergo osmotic lysis. And you'll get a pink solution when you spin the cells down. That pink solution is almost pure hemoglobin. So you can study hemoglobin. The choice of problem is very important. I show this to you only because people in India, very often I find, I am now quite old and I've been associated, everybody complains about money. Sometimes I would like to see people who complain about the lack of ideas. And Pauling, of course, here writes this letter here to Millikan, the famous Millikan of the oil drop experiment, which some of you might have read in high school physics. And he talks about spending what I call unspent balance. Unfortunately, the Department of Biotechnology and the other departments which fund your research, their finance departments don't like all of this. But Pauling even then was writing, and I found this in the Pauling archives, wonderful. Would it be possible for me to use an advance of $250 if necessary from the next year's grant? Today, you try asking this with the director of an institution, and I've been one. I know the kind of answer that you will get. It will invariably be no. What did Pauling want to do? What Pauling wanted to do was apply a new physical method which has come. Tiselius method of electrophoresis for separating proteins on the basis of their charge. And he wanted to study sickle cell hemoglobin. This was an inspired choice of problem. I show you the Tiselius diagrams, what are called Logsworth-Cunning diagrams of normal hemoglobin, sickle cell anemia, and you can see that the band has shifted. The position on the gel has shifted. That means the protein has a different charge. Then he did the remarkable experiment, the control experiment, which many times students will not do. He mixed the two samples together and put them back onto the machine. And then you see he got two bands. So it was not an artifact. And then the most inspired experiment of all, he got a person who was a carrier of the sickle cell disease, but did not suffer from the sickle cell disease. One abnormal gene, one normal gene inherited from the parents, and then the sickle cell trait, it resolved into two bands. So there were two kinds of hemoglobin there. This is the conclusions of the Pauling paper of 1949. Alterations in protein structure, hemoglobin, lead to disease. Disease is inherited, sickle cell anemia. Hence, genes precisely determine the structures of proteins. He also anticipated the one gene, one polypeptide hypothesis from this results. Beadle and Tatum, he was aware of their work, and it turns out that Pauling's paper also reached to this conclusion. Avery published his paper in 1944. He'd done his work a little bit earlier. In 1949, DNA was not yet accepted as the genetic material. Vernon Ingram, who established this mutational difference, published this paper later in 1957, where he used peptide chemistry really here, hydrolyze the protein, separate the peptides on paper chromatography, and establish the site of mutation in the protein. Here, the site of mutation was established in the protein long before the site of mutation was established in the gene. This is because Sanger invented protein sequencing first before he invented DNA sequencing. Then Ingram says here, thus it can be seen that an alteration in a Mendelian gene causes an alteration in the amino acid sequence of the corresponding polypeptide chain. And this is, of course, the first connection between the chemistry of polypeptides and Mendelian inheritance as far as disease was concerned. Today, of course, we know that it is a single point mutation, but Ingram anticipates this. He writes in his paper, it is likely to be a single point mutation because 1957, remember that the genetic code was still not established. But in 1957, Crick's ideas, and I will come to them towards the end, were known that there was a genetic code. Pauling commented on this later, and that is the basis of our current understanding of the molecular basis of disease. He says it is astounding that the difference in structure is so small. A change from valine to glutamic acid. Only about a dozen atoms out of 10,000 the molecule are different. On such small atomies, man's fate depends. We've always believed that man's fate depends on Mendel's laws. Genes are the carriers of information, but genes transfer that information to proteins. Proteins, as Arthur Kornberg said, DNA merely writes the script, the proteins do the acting in the drama of the cell. This is Avery's work. Avery's famous paper in the Journal of Experimental Medicine in 1944. But Avery was a very, very cautious man. He was a small man, a very shy man, and one of the group photographs at Rockefeller, which I saw, doesn't even give where Avery is standing. It merely says he was always the shortest person in the room, being only 5 feet 4 inches tall. But he was a meticulous scientist. You should read the 1976 book. It is called, it's called The Professor and the Institute, and DNA, I think. Professor and the Institute and DNA. This is written by René Dubos, who was Avery's student, but himself the man who first discovered the first antibiotic, gramicidin A. The antibiotic era does not begin with Alexander Fleming, that is legend. The antibiotic era really begins with Dubos and gramicidin, because gramicidin was clinically used for external wounds because of its antibiotic properties. He says that the major interest has centered in attempts to isolate the active principle from crude bacterial extracts and to identify possible its chemical nature, or at least to characterize it sufficiently to place it in the general group of known chemical substances. He's a very cautious writer. What I've written there in yellow says, instead of claiming something, today, of course, you can't publish a paper unless you've made a claim in the title, another one in the introduction, and sometimes a different one in the conclusion. If the results of the present study on the chemical nature of the transforming principle are confirmed, then nucleic acids must be regarded as possessing biological specificity, the chemical basis of which is as yet undetermined. But then we have to ask, where did DNA come from? DNA first came from the laboratory of Friedrich Meister at Tübingen many, many years earlier in the 19th century. Meister, working in the physiological chemistry laboratories of Hauptzeller, actually did this, and that's Meister's first paper. Today, at the University of Tübingen, you have these plaques to commemorate the discovery of DNA. Meister discovered DNA. Avery established DNA as a genetic material, and Watson and Crick then gave you the structure of DNA. In between, of course, there is the story of the chemistry of the nucleic acids. For a long time, the chemistry was worked out by Levine, again at the Rockefeller Institute of Medical Research, now the Rockefeller University in New York. He, of course, worked out the chemical structure of the nucleic acids correctly. But he thought that they are just a repetition of a tetranucleotide, and therefore information could not be repeated in such a sort of monotonous molecule. And the tetranucleotide hypothesis, whose centenary has now been sort of celebrated in 2009, it appeared in 1909. This is chemistry moving along. It did not really delay this. Subsequent historians have said this. To some, this hypothesis was a major obstacle. To others, Levine's discovery was a milestone in the quest for uncovering the chemistry of life, because he worked out the detailed chemistry. There was, of course, Chargaff, ignored and never made a secret of the fact that he was ignored in the books that he has written, in the essays that he's written. He's the one who gave us the equivalence principle between the bases, A is equal to T, G is equal to C, absolutely fundamental to the Watson-Crick structure. He's written a book called Heraclitan Fire. You have to use a dictionary very frequently to try and understand what Chargaff says. And he is, of course, a very unhappy man. Then there is the Watson-Crick double helix. You could see that the famous first paper of the Watson-Crick double helix, except for its last sentence, has very little information. It has a picture like this. This is the kind of cartoon that you see today in school books. That's about it. The other structure that you see is much later, the photograph. But the Watson-Crick structure is really based on diffraction data. The X-ray diffraction data are the data on the left and the data on the right. The Wilkins paper, all these three papers appeared one after the other in the April 1953 issue of Nature. The diffraction data on the left is Wilkins data. The diffraction data on the right is Rosalind Franklin and Raymond Gosling's data. That is the famous photograph called Photograph 51, after which now I think there is both a movie and a play. That is what the structure of DNA is really based on. All the technical information which is necessary to build the Watson-Crick model is there, what I've highlighted in red. There's almost nothing in the Wilkins paper. Yet in 1962, it is Crick, Watson, and Wilkins who shared the Nobel Prize. Rosalind Franklin, of course, had died by then, but she was never in contention for the Nobel Prize. She's now, of course, posthumously been recognized as one of the most famous examples of gender discrimination in science because of the kind of portrayal that Watson made in his later writings. But Crick, of course, was a remarkable man. Crick foresaw biology, and he was a physicist. He was a physicist trained in diffraction physics. He foresaw biology. And then he knew that the main function of proteins is to catalyze reactions. He also realized that the main function of genes is to produce proteins. And I would show you this paper of Crick on protein synthesis. This paper, written in 1957, is something that everybody should read. This is a 1957 symposium. As yet, the code is not completely broken. Kornberg's work has just appeared. Korana's work is still in the future. Nirenberg's work is still underway. And Crick anticipated everything that we know today about the transmission of information from DNA to RNA to protein, the three-letter genetic code, and so on. And he actually concludes in this paper that for both these reasons, I shall be surprised if the main features of protein synthesis are not discovered within the next 10 years. And they were indeed discovered within the next 10 years. There are three pillars of modern biology: Darwin and Mendel, which have already come on my first slide. I've inserted the third pillar now, that is chemistry. This is Avery's establishment of DNA as a hereditary material, the detailed chemistry of DNA, beginning with Misha, going through Levine, ending with Chargaff, which led to the Watson-Crick structure. Remember, the Watson-Crick structure is a chemical structure. It's a three-dimensional model in which the base pairs hold hydrogen bonds. Pauling was one of the men who completely popularized the idea of the hydrogen bond in protein structures. It is hydrogen bonding which holds the structure of DNA. The 3.4 angstrom separation between the bases is the same spacing that Rosalind Franklin sees in her photograph. That is the stacking of the aromatic rings. The DNA structure is a chemical structure. It's a chemical molecule. And therefore, there is no ignoring the fact that mutations are chemical transformations. The DNA repair is one in which enzymes now repair the DNA. In sort of coming toward the end of my presentation, I go back to Darwin. You can't write biology better than the way Darwin wrote it towards the end of this book. He says that there's grandeur in this view of life, with its several powers having originally been breathed by the Creator into a few forms or into one. He's a religious man, and that while this planet has gone cycling along to the fixed law of gravity, there is the allusion to Newton. From so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. It is this variation. And how does this variation happen? How does this information get transferred? How do biological organisms get selected? What are the environmental pressures that drive mutation and Darwinian selection, which are important? One of the famous evolutionists, John Maynard Smith, put this very well at the level of proteins. He said, for example, the concept of natural selection can be integrated with the concept of a protein space, a sequence space. And he gave this example. Take the word word. Word means something. 4 letters. Mutation, war, still means. Gore, still means something. Gone, another meaning. Gene, another meaning. So when we look at the complexity of biochemistry, today what do we say? Even the simplest of bacterial cells, although you are all in agriculture, I must remind you that the origins of life on Earth are really with archaebacteria and bacteria. Microbiology precedes anything else. Plants came so much later. The biochemistry of plants is a derived biochemistry, a biochemistry in which evolution has already had its role as bacteria and archaea evolved over 2.5 billion years of Earth's existence. Therefore, protein evolution is best understood by going back in time. One last look at Avery before we end. Avery anniversaries have been celebrated, and there can't be anything better written than what Lederberg, of course, was after a long time, after Müller, Lederberg was a man who called himself a geneticist, albeit a microbial geneticist, if not a plant geneticist. And he said in his Nobel lecture of 1959, he said that genetics should now be recognized as also timely. He did not talk about his own work. In his Nobel lecture, he talked about Avery and the foundations of biology. And he just says, the precise demarcation of genetics from biochemistry is already futile. But when genetics has been fully reduced to its molecular foundations, it may continue to serve in the same relationship as thermodynamics to mechanics. Today, genetics and biochemistry cannot be separated from one another. The coordination of so many adjacent sciences will be a cogent challenge to the intellectual powers of our successors. And that's what he said in 1959. You could go back and look whether this has been reflected in the 60 years after Lederberg said this, in transforming the way a biology education is done in India. The first title that was suggested to me by the organizers was to talk about something educational. I'm, of course, incompetent to talk about that because I don't teach genetics. Chemistry, of course, Kornberg said, is the lingua franca of medicine and biology. Biology is the foundation of both medicine and agriculture. And none of it is possible without the intervening chemistry. Finally, I will conclude with my favorite quote, and that simply is this, that what scientists do, irrespective of the discipline in which they work, they try to understand nature. The physicists try to understand nature in their own way. The geologists understand nature in their own way. The cosmologists do too. Chemists, physicists, biologists, all of them together in understanding nature. Why do we try to understand nature? Because science is the study of nature. It's nothing else. The best definition of science I can see is science and study of nature. Science is not there to solve everybody's problems. The problems get solved as we go along because we have understood something in nature. We can't also always transform nature. Sometimes science is thought of as a discipline in which we exploit nature. If we exploit nature too much, we end up with the kind of problems that we have. Today, science must also be sustainable. And therefore, even when talk about recombination, the recombinant DNA, genetically modified crops, solving food security problems and so on. Remember that you're also only addressing human problems. But human beings are not the only inhabitants of the earth. There are lots of other creatures, including microbes, who will outlive us. And therefore, one must be thinking about the environment and ecology as a whole and ask what damage one might do, or how does one even recognize it. The antibiotic experience has taught us this. The antibiotic experience, for example, now has taught us that resistance is the problem that everybody is worried about. For example, in dealing with an old disease like tuberculosis, which appeared to have been completely eradicated. So biology in its manifestations is a very vast subject. We study nature. Goethe said we are surrounded and embraced by her, powerless to separate ourselves from her, but powerless to penetrate beyond her. You can't define nature better than this. And this is a translation, not by me, but by Thomas Huxley, writing the first editorial in volume 1, page 1 of Nature, 1869. Lastly, my acknowledgements to the two institutions, the Indian Institute of Science and the National Center for Biological Sciences, which have tolerated me and provided me a home for much of my professional life. Thank you very much.
Professor Balram, maybe there are one or two questions. So is there a question? I would like to, because with such a illuminating lecture, many things will come to mind. Just to, yes, please. Please speak loudly or give the mic.
Sir, since Mendel, we have traveled a very long road of science and academic development. My question is at the second part of the road. In the last 1 decade, we have seen two very powerful techniques making round throughout the world. CRISPR-Cas and artificial intelligence. Both they are mutually exclusive, applicable, and extremely dangerous in the abuse and misuse and proper use. And I can see it coming that they are the two powerful nails in the coffin of the end of humanity if the scientists are not serious in its proper use. What is your take on this? Thank you very much.
First of all, I would, when you talk about these two, artificial intelligence and the CRISPR-Cas9 gene editing methods, remember that these are now new technologies. And many, many years ago, in reviewing a book on DNA, the theoretical physicist Freeman Dyson said that science is often driven by new technology rather than by new concepts. And this is true. Much of our science has been driven by new technologies. Molecular biology is the best example. And sometimes these technologies are remarkably useful, like, for example, the RT-PCR technologies. Both the discovery of reverse transcriptase and the discovery of the polymerase chain reaction put together give us this wonderful technique. But many times also, like in the case of gene editing technologies, you are now trying to interfere with a natural process. You might do it very well. You have a specific reason for it. In medicine, there is a very specific reason to try to correct some kind of inherent genetic defects if you can. But many times you can't anticipate the results of these technologies. I'll come to artificial intelligence in a minute. I'll give you a longer answer. I'll give you another example. You know, chemistry is, in the year 2000, the American Chemical Society asked, who are the most influential chemists of the 20th century? And so all chemists voted. So I'll tell you the names of the first three people. The names of the first two people were known to all chemists. Pauling was number one because Pauling had revolutionized everything. R.B. Woodward, who revolutionized organic synthesis, was the second, because he was a hero to every organic chemist in America at that time. But the third person was a complete unknown to most of the people, but he was the man who invented plastics and polymers at the DuPont company. But it's plastics and polymers which really drove much of our progress in the 20th century. Today, what are we doing now? We are now passing legislation. We are worried about environmental, we are worried about things which are, the same thing is true with DDT. DDT received the Nobel Prize almost immediately after it was found. It had applications in agriculture, it had applications in medicine. You killed mosquitoes, so you got rid of malaria. You killed pests, so you helped your plants. But eventually, what are we doing? We are now worried about other things, about pesticides and so on. So I think science and technological advance is a double-edged sword. The classic example is atomic energy. It has fueled our fuel. It has been a major fuel for fears among human beings rather than fueling economies. And we don't use atomic energy that much in power generation. So I think one needs to be balanced and careful. Sometimes scientists oversell. There's always a nexus between scientists and politicians and administrators in America here in driving things forward. And once somebody is told that this is the best thing to do. Artificial intelligence is one such thing, which is, artificial intelligence is not a panacea for everything. If anybody thinks that artificial intelligence is going to replace human intelligence, you'll have to wait for a long time. It's by and large developed from pattern recognition and the ability of machines to learn to recognize patterns. It has very useful applications. In medical diagnosis, it's got very useful applications because you can get diagnosis done where there are no doctors in rural areas and so on. It may be very, very valuable. We can't expect it to do everything. So I would still say that the jury is out on how much the CRISPR-Cas9 technologies are going to revolutionize your life and mine. Probably not. Looking at you and looking at me, I don't think it's going to revolutionize our lives.
There is one stupid question in my mind, but maybe I can ask if you allow. Okay. The question is that we always learn that the principles of physics, chemistry, and biology are working in unison and in harmony. Are there examples where there is a conflict in these principles? And if there is so, how is it resolved?
As far as I know, there are no biological phenomena which are in conflict with the principles of physics and chemistry. Where they appear to be in conflict with the principles of physics and chemistry, it is only because you haven't thought about them deeply enough. In fact, at the National Center for Biological Sciences that I have, there are a group of young and not so young, I have to say, actually, here, my definition of young is getting higher and higher, but this is relative. Scientists who are physicists who are working in biology, and they're actually funded by a foundation which gives money only for work in mathematics and in theoretical physics. And they look at biological phenomena. Many of these in cell biology, which you observe. And I guess for every new phenomenon that you observe, there is an explanation. You only have to look for it, and it requires very good physicists to find it. There's one which I can't explain because I haven't understood it, but I sent it to one of these people. Appeared, I think, in last week's issue of Nature. There's a phenomenon of elasticity which they're looking at, but I think almost everything. Part of the reason is, just think about it. Some years ago, the Bombay High Court had a judgment. It said that steam is not a chemical. And the High Court say all these things, but it said steam is not a chemical. So industries who have litigation have to listen. But steam is a chemical. It's just another form of water. So the Royal Society of Chemistry in London offered a reward of £1 million to anyone who would bring a material which is non-chemical. Now, the only people who give you non-chemical chemicals are Patanjali and their equivalents in the West, and also sometimes in our own cosmetics industry. Now, if you think about it, there's nothing around you which is not a chemical. The sand, everything so chemistry is all pervading. And if the interactions of atoms with one another are determined by the laws of physics, large collections of atoms cannot be different from smaller collections of atoms. There'll be only a few new laws to be learned. So I think we can rest assured that for the foreseeable future, I don't think a revolution is going to happen.
So, friends, I'm sure you have heard a very illuminating lecture. It has been kind of a relearning the whole process in unison, without any conflict, as I was trying to understand myself. And therefore, we once again clap for him and for this nice lecture. Our next speaker is Professor P.K. Gupta. Professor Gupta is a real teacher. And why I say so, because I know that here in India, he has trained almost 3 generations of geneticists. And when we were students, his group was interested in mutation breeding and particularly legumes. And in 1996, we all gathered to celebrate the retirement of Professor Gupta. And we did not know at that time that even before retirement, he has reincarnated into a molecular biologist and the person who will be working on genomics. And for next almost 26 years, after his retirement, he has rigorously worked in the area of plant genetics, starting from quantum genetics and also the genomics, and has contributed tremendously to the genetics of wheat and also a large number of others. So that is what is Professor Gupta. I have been fortunate to learn from him, and I have also been fortunate that I do not fear him and frankly ask so many questions from him. So Professor Gupta, this is your turn.
After my lecture. All right, no issue.
Friends, it's a great pleasure for me to be here. I always enjoy talking to students and colleagues, and particularly in Indian Agricultural Research Institute. Whenever Ashok invited me, I never said no. So 2 months back, Ashok wrote to me that I should deliver a lecture at this particularly important meeting. And he gave me a title, which is similar to this title, but it was something like Mendel to computer science and Qtls and so on. But at that particular time, I was busy in writing and getting an article published in Trends in Genetics, the Earth Biogenome Project. Therefore, I asked him to change the title, and this is why this title is like this. But you will see in my presentation that only 3 slides are devoted to Earth Biogenome Project. The rest of the presentation will be overall genetics. Because the genesis of this lecture, I will start with Mendel. I will not talk about pre-Mendelian work. And starting from Mendel and also the biometrical or quantitative genetics by Galston and his group, I will then start the history of genetics rather quickly, and about half of the lecture or 2/3 part of the lecture will be devoted to what has happened during the last 120 years. But 1/3 of the last presentation will be devoted to a part in which I have been excited during the last 2-3 years, because there are new things emerging everywhere, and I always enjoy reading and writing about something which is exciting and something many students don't read, and therefore I would like these topics to be known by these students, and therefore I read and write. So starting from this particular slide, we have been talking about that the 200th birth anniversary of Mendel is being celebrated all over the world. There are a large number of journals which are devoting their special issues to this particular celebration. You can see there are conferences. As Bruno, Jack, we have been talking about it. There are special issues of plant cell, heredity, and several journals. Okay. And there is an editorial in Nature which I enjoyed reading, which talks about eugenics. Therefore, you will see that 2 or 3 slides I will also talk about eugenics and its importance in the present context. Then there is a recent article in Nature Genetics on July 11th, which talks about the history of Mendel's laws and the controversies which we yesterday also talked about. Dr. B.D. Singh talked about it. And then there are 3 articles in Nature Review Genetics, which also appeared in July 2022. And recently I saw that the cover page of Genetics, published from USA, is also devoted to Mendel, showing the pea plant on which Mendel worked. You see, this picture shows 3 important scientists who worked after Mendel and devoted their time in some disagreement with Mendel, not really contradiction. They worked on human genetics, and while studying some of the traits in human, like tallness and also intelligence, they found that Mendel's laws did not hold good in explaining the inheritance of human traits in those particular cases. And therefore, sometimes in our textbooks also we say Mendelians and biometricians. There was a controversy for some time, which was later on resolved. But you will see, I have shown this picture also because I'm going to talk a little bit about eugenics, which was proposed by, yes, I think that's better. Morning. Oh, hello. No. you see, these three scientists applied statistics, tried to explain plant height, inheritance of plant height and intelligence on the basis of regression. But one of the most important papers which I am going to talk about later on after eugenics was proposed by Carlton in 1980s or 1990, and he mentioned in his publications, as you will see, that eugenics should be used for improving human species by selective mating, and also for breeding out diseases, disabilities, and other undesirable characteristics. And therefore, during the first 2 decades of the last century, there were arguments in favor of eugenics, which was practiced in some parts of the world. You can see that in Germany also, and this was practiced against Jews, for example. And then most recent incarnation of eugenics is in the form of white replacement theory, which they have talked about in editorial, in Nature editorial also this is mentioned, where shooting incidents have been correlated with killing of blacks in USA. They have described it as white revolution theory. There is a particular incident, recent incident of shooting in USA, where 13 people were killed, and 10 of these 13 people were blacks, by an 18-year-old boy. And he also pasted a manifesto, 29-page manifesto, which starts with this phrase, kill as many blacks as possible. So these young boys and persons are obsessed that whites are being replaced by blacks and Asians, and therefore genetically the race of whites is being diluted. And therefore they talk about this practice of eugenics, although we cannot say it in large measure, but the incidents have been correlated with this. And you can see that therefore there are demonstrations against these shooting incidents in USA. I have taken this picture from Nature, recent issue of Nature. Then after this brief introduction about eugenics, I also like to talk about some Mendel's work, which is not really Mendel's laws. 1965, he gave his work on P, but after 1965, he started working on several plant species, 26 different genera. 26 different genera he used, including Heratium and maize, where he tried to find out whether what he observed in case of P, whether it is found elsewhere also. And in several letters, there are 10 letters which are available, which he wrote to Carl Nageli. Carl Nageli was a professor at Zurich, later on at Munich, and Mendel thought that he was a very intelligent person. Therefore, all the results which Mendel was getting, he was communicating to Nageli for getting advice. And in these letters, Mendel always addressed Nageli as esteemed sir. It means he really had a great respect for this man, and expecting that Nageli will give him correct advice on the results which he were obtaining. And Heratium was selected as one of the most important material for further investigations, because Heratium had a lot of variability in nature, and therefore Nageli also suggested him that you should work on Heratium, knowing very little that Heratium is an apomate. That was the problem, and therefore in several publications, several letters which Mendel wrote to Nageli, he is sending him the results on Heratium, where he says that in F1 I am getting variability and not uniformity as expected in case of P, and in F2 he was not getting variability which he was expecting, segregation, and this was really puzzling for him. But the beauty of these letters is that the results have been communicated to Nageli in exactly the same form, number of seeds, number of plants grown, in literally exact terminology, and no manipulation was there. And he also mentioned in several of his letters that the laws we discovered in case of Pisum do not hold good universally. There must be other laws of inheritance, other methods of inheritance for explaining the inheritance in many. So you can see that Mendel never emphasized that the laws of inheritance which he proposed will be of universal application. He also worked on maize, and this is particularly mentioned in a review which Rhodes in 1984 wrote in Annual Review of Pathology, where he is mentioning about Mendel's work, and he mentions that Mendel was the true first maize geneticist. And Mendel worked on maize, and there also he had some difficulty because while working on certain seed traits, he did not get the desired results because of the phenomenon of xenia, which was not discovered till then. In the F1 plants, the parental seeds which you obtained showed the segregation which in Pisum you would expect in the next generation. So because of this phenomenon of xenia, he had problems, and therefore he could not explain these results also. But in this paper, I read that Rhodes' paper says that Mendel was really the true first maize geneticist in two cells. This is another very important paper which I read when I was writing my book on quantitative genetics. Weldon's paper. I was not aware about this paper earlier, but Weldon wrote a very comprehensive paper, 100 pages in 2002. And in this particular paper, he describes his results, which he obtained by collecting pea plants, pea seeds from the market of a large number of varieties. And then he tried to find out the variability for the traits, which Mendel studied. And he found that the two extremes which Mendel used, they were not really available in the seed collection which he obtained. And there was variability. You can see wrinkled seeds to round seeds and green seeds to yellow seeds. There is great variability, and he says that there is continuous variation in the traits which Mendel studied. And therefore, he calls it alternate inheritance. He does not say that Mendel was wrong, but he says there is an alternative mechanism of inheritance which shows continuous variation. And in order to explain this result, we can perhaps conclude that Mendel used the extreme types, which were exhibited because major genes, and therefore there is no contradiction. But in one of the editorials which I read in 2016, when Mendel's 150th year was being celebrated, there they say that if Weldon died very early after publishing this paper 2004, and according to this editorial, he says that if Weldon had survived, genetics would have perhaps taken another course because there would be emphasis on quantitative genetics then. But we'll see later on also, although quantitative genetics could have taken this new turn because of the emphasis by Weldon, but the techniques were not available to understand this quantitative genetics. And therefore, even if Weldon had survived, the analysis of this continuous variation was not possible because the techniques were not available. Then you see, after 1900, one of the major important area for about 20 years or 30 years was the preparation of genetic maps. And one of the important points which the students, I want to tell the students of genetics is that for preparing the genetic maps, we always try to explain to the students that the genetic distance between any two genes is directly proportional to the recombination frequencies. This general principle that genetic distance is controlled, is dependent upon recombination. Recombination frequency depends upon genetic distance, holds good at very low distances, small distances, but as the distance between the genes increase, then this relationship does not hold good because the recombination frequency does not increase proportionately with the increase in the size, in the distance between the two genes. And you can see that at 50% recombination, there is no further increase. Therefore, for preparation of genetic maps, mapping functions were proposed. One was proposed by Haldane. He did not take into consideration the interference, and the other was proposed by Kosambi, which took into consideration the interference also. And most of you may be familiar that in majority of the softwares which are used for preparing genetic maps, Kosambi's mapping function is used throughout the world. Also, we also know, as some of you may be familiar, that recombination rate does not proportionally increase with distance, and also recombination rates differ in different regions of the chromosomes. One of the scientists from USA came and delivered a lecture in our department where he showed significantly, particularly in case of wheat and maize, that recombination rates, not only they do not show a relationship with the increase in size, but they also differ in different regions of the chromosomes. And therefore, you cannot generalize that you can conclude the distances between the genes on the basis of recombination frequencies. Another important part of this map function is that intragenic crossing over was not known till about 1940. In 1940s and 50s, for the first time when I started teaching genetics, I discovered that there is crossing over. Genes are not really point mutations, points. They cannot be represented by points, but they have dimension. And therefore, intragenic crossing over could be discovered. Mutational sites within the gene could be discovered, and mutational maps, micro maps could be prepared in large number of cases. And that gave rise to the fine structure of the gene, which was in R2 locus. We discovered that the R2 locus consists of two different cistrons. Many students do not really understand the meaning of cistron. Sometimes they say that cistron is a gene, but this is not really true because a gene can have more than one cistrons. Then induced mutations. This was another important, very important area which started in 1927 with Muller, and then Muller used Drosophila and showed that X-ray can create mutations. Stadler used maize and barley, and then Auerbach later on used chemical mutagens. And these three groups started a new field of genetics, and particularly because in majority, when you study the traits, you need alternative alleles. And for majority of traits, you may not have the mutant allele, so that you cannot study the genetics. And therefore, in order to make it possible to study genetics, mutations were necessary, and therefore techniques were developed in order to create variation so that the genetics may be studied. Then starting from 1940 to 1970, there was a very productive period of biochemical genetics and molecular genetics. And the biochemical genetics work was started with Biedl on the Neurospora, and one gene, one enzyme hypothesis, and then one gene, one polypeptide hypothesis. They were proposed at a particular time. DNA is the genetic material that was discovered by Avery, as mentioned by Balram also. And DNA double helix was discovered by Watson and Crick. Discovery of transfer RNA was done in 1958. Discovery of messenger RNA and protein synthesis in 1961. And this led to the proposal of the mechanism of protein synthesis in 1961, when I just started my teaching career at Gorakhpur University at that particular time. And then simultaneously in genetic code, I still remember in 1960 when I was just started teaching at Gorakhpur University, there was a headline in the newspaper, genetic code cracked. So that was the paper by Nierenberg at that particular time, when the first codon AAA was discovered to code for phenylalanine. And then there was another important discovery of course, after this, there was a Hawley. Hawley discovered the sequence of alanine transfer RNA. And this sequence, which was discovered by Hawley, who also shared the Nobel Prize, was utilized by Khorana in order to synthesize a gene. synthesis of East alanine transfer RNA, 1970 paper. And before he published this paper, Khurana realized that the gene he was synthesizing was not really the complete gene. It was the ultimate product. And there are precursor transfer RNAs which are much longer. And therefore, later on, Khurana synthesized another gene which was much longer than the gene which he synthesized in the beginning. Then there was a very productive period. It started from late 1960s when it was discovered that the nucleic acid or the DNA in the nucleus consists of two important fractions. There is a lot of repetitive DNA. There are, and this graph I have taken from a paper which Chitranjan Bhatia published in 1973 in Wheat. And I remember when I visited UK in 1974, everybody was asking me if I know Chitranjan Bhatia, because it was very surprising for them that such a work could be done in India. Then another important aspect was the sequencing of protein molecules, which led to the sequencing of insulin molecule, which has A and B genes by Frederick Sanger, and then DNA sequencing, which led to the sequencing of 5X174. And you all know that Frederick Sanger got two Nobel Prizes for these two important discoveries which he made. Along with this, there was split genes, which were discovered in early 1980s. And we came to know that the gene is not continuously coding. There are exons and introns within the gene, which take part in protein synthesis. Then the period comes, this 1980 to present age, where I have been involved during the last 30, 40 years myself. I learned this technique of molecular markers in Canada during my visits, and then of course used it in my own lab later on. 1980 was the beginning of the availability of molecular markers in the form of RFLPs, as you can see, then RAPDs, then SSRs, AFLPs, and you can see that I had to change this chapter in my book regularly because there were always new molecular markers developed almost every 5 years. Then in 2000, SNPs were developed. I had to write a review in Current Science on SNPs. Then DART markers in Australia were developed, which became very popular later on. And more recently, after 2010, when genome sequences became available for a large number of plants and animals, we came to know about these CAVs and PAVs, and then structural variations and Kamer variations. Particularly these two last molecular markers were discovered only recently and are being increasingly used by many workers. I wrote a small article in Intraogenic Genetics on these, and then also in bioassay. And then I had to, because of these developments, I had to revise my book on cytogenetics. And now my book on cytogenetics has two parts: classical genetics and molecular genetics. Part 2 deals with molecular cytogenetics, which has about 12 chapters. With the help of these molecular markers, such synthetic collinearity maps could be developed in related species. Sequences could be compared. And this particular popular map was designed in Norwich by Gale at that particular time and his group. You can see the outermost ring is oats, then TTC, including wheat, then maize in 2 circles, because maize is considered to be a tetraploid. And then you have sorghum, green color here, sugarcane, and then foxtail and rice. The genes on the chromosomes of all these different grass species, they were collinear in their arrangement of the chromosomes, and it could be shown very clearly, showing conserved linearity. And students may be familiar that there is a difference between synteny and collinearity. I will not go into the details of this, but those students who do not understand it, they should go back and find out the difference. Molecular markers for Qtls. This is something which started in 1989. And I tell my students there were two important developments which led to QTL analysis. One was molecular marker development, the other is statistical development, statistical genetics, where this paper, Lender and Botstein papers in 1989, was a landmark paper which suggested the technique of interval mapping, which could be possible only with the availability of molecular markers, because if molecular markers were not available, QTL interval mapping will not be possible. Then there was another technique which was pioneered by Ed Buckler at Cornell. Starting in 2001, candidate gene approach was used in case of maize. And in 2005, large number of papers during 2005 and 2007 were published utilizing association mapping, genome-wide association mapping. And therefore, this field became very, very popular. You can see in this particular diagram that interval mapping depends upon development of a mapping population. And this mapping population will be used to scan every interval between any two adjacent markers for the presence of a QTL, and a LOD score, high LOD score, will show the presence of a particular QTL at a particular place, and therefore you can identify the flanking markers and utilize them for a variety of purposes, for cloning of the QTLs and also for plant breeding. In case of GWAS, we use linkage disequilibrium, and therefore we can use a large number of lines, genotype diverse lines, and these diverse lines germplasm used for genotyping and phenotyping. And genotyping, after genotyping, we can use candidate gene approach, or we can use whole genome association mapping, which leads to identification of marker trait associations. Many people call them QTLs, but I prefer to call them marker trait associations, because you are not identifying genes, you are only identifying the markers which are associated with a particular trait. And Ed Buckler, of course, was the most important person. He continues to be the most important person in this area at Cornell, and everybody looks forward. I had a chance of listening to him in China, and then also he visited India also. Then with the availability of these two techniques, interval mapping and GWAS, it was possible to identify markers which became available and utilized for marker-assisted selection. Ashok himself has done important work in case of rice, utilizing molecular markers. Prasanna again has used a number of techniques. But the important point which I am trying to make here is that majority of the traits which have been handled by molecular marker-assisted selection were qualitative traits in most of the cases, and it was not possible with the availability techniques to utilize this particular technique for quantitative traits controlled by hundreds of genes. So complex traits remained a challenge. Although marker-assisted recurrent selection and genomic selection could be used for these complex traits, and these traits are still not very widely used, although they are being practiced in several countries. In our countries also some effort are being made, but these are the two techniques which can be used and will be used in future for study of the genetics of complex traits. QTLs for genomic selection, this is something new which I read during the last few months, where you can use QTLs, not molecular markers. You can use QTLs for genomic selection rather than molecular markers. So you can combine the QTL analysis with genomic selection and then not utilize all the markers, but only selected markers associated with QTLs for the purpose of genomic selection. This is a scheme for genomic selection where we have a training population, where we try to train the population, which is a part of breeding population. And with the help of this training population, when it is trained and standardized, then we estimate the genomic assisted breeding values, and these breeding values are then used in the breeding population, where without phenotyping, you can select the plants which are superior. And this technique is being utilized at several places, more so in animals than in plants, but it is yet to be standardized because the prediction accuracy of this particular technique is still about 50–60% in majority of cases. Then comes the period of genomics, 1995. This period started, and there were two techniques available for genomic sequences, the clone by clone sequencing, where we first prepare the physical maps and then utilize the clones mapped these clones for sequencing, and shotgun sequencing, where we do not prepare a physical map but utilize the short segments for sequencing. And both these techniques are used, but this technique, shotgun sequencing, has become more popular because it saves time, and although more statistics is needed in this particular technique. And you can see that the next slide will show this is the first genome which was sequenced Haemophilus influenzae. The paper was published in Science, and this was followed by sequencing of a large number of organisms. This is Arabidopsis, which was published in December 2000, and this marked the beginning of an important discipline, in my opinion, which we say structural variations and ancient polyploidy. I have devoted 2 chapters in my book on this. Structural variation means that with the help of this particular technique of genome sequencing, we could discover that even in diploid crops, there are extensive duplications and deletions and translocations at the molecular level, which could not be studied at the chromosomal level. Majority of these structural variations, which were and polyploidy, these are those changes which could not be studied at the chromosomal level by classical genetics, but could be resolved by sequencing. And then you see, initially we started with sequencing only a single genotype of a particular individual in order to find out the sequencer. But soon it was realized that when you sequence only one particular variety or genotype, this does not sample the entire variation, and therefore you must sequence a large number of genomes, large number of genotypes of the same plant in order to study the variation which is found within the species at the genome sequence level. And therefore 3 types of sequences were discovered: core sequences, dispensable sequences, unique sequences. Core sequence will be present in all the plants. Dispensable sequences will be present in some and not others. And then unique sequences will be present only in individual plant and not in any other plant. So these 3 types of sequences could be discovered. And in large number of species, we have now sequenced large number of genomes. For example, in case of rice, 3,000 different genotypes by IRI sequenced, and Rajiv Varshne at Ikisat sequenced large number of chickpea genotypes. From sequencing to post-genomic era, you see, when sequencing of human genome was completed in 2003, then there was an announcement that we need techniques which will be cheaper and will give the results very, very quickly. And therefore, there was a competition for developing these techniques, which were described as next generation sequencing techniques. And between the year 2005 and 2008, a number of sequencing technologies, next generation sequencing technologies were developed. Initially, Sanger's DDFC and nucleotide technique was popular for about 30 years. There was no other technique which was used. But following this, in 2005, these techniques were developed, 455, Illumina, Solid. But one of the problems in these particular techniques, which were initially developed, was that only small sequences up to say 60 nucleotides to 100 nucleotides could be sequenced, and therefore statistical exercise was rather vigorous in order to assemble the genome. And therefore, more recently, after that 2015 or 16, these 2 technologies, biosequencing and nanopore sequences, they took over, because in this case you can sequence long DNA sequences of more than 1,000 base pairs, and therefore majority of cases these biosequencing and nanopore sequencing are being used in order to refine these sequences which were earlier available. And then this was also followed simultaneously in parallel: genome versus transcriptome, exome, epigenome, metabolome, microbiome, and spatial genomics. I will come back to that later on, a little bit later on. Spatial genomics is a recent topic, which means that the sequences will differ not only between the individuals, but also between the different tissues of the same individuals. and also between the different cells of the same tissue. So this is remarkable. This is, they call it spatial genomics, therefore. And sometimes we call it fourth dimension, 3-dimension, and then fourth dimension. Then gene editing technique. You can see the different steps which are involved. Gene targeted, one of the most important points which the student should remember, you can only edit a gene whose sequence is already known and the nucleotides involved in allelism should be identified. So you should know the target selection, 20 base pair upstream of, there is another limitation of this, proto-spacer adjacent motif should be present, 20 base pairs downstream, only then you can edit according to this particular sequence, this Casper edit technique. And then following this assembly of SG RNA, single guide RNA and Cas9, you can do the gene editing, transform the plant, regeneration and screening of the plant, and then selection of the edited plants. And when you select the edited plants, a very small frequency of plants will give you the desired results. That is one of the limitations of this particular technique. These are two important examples of gene editing, which are popular in the literature. One is white mushroom, the other is maize, drought tolerance waxy maize, which have been commercialized. Then there is another recent example of gene editing in tomato, which is published in 2022, where tomatoes rich in vitamin D3 have been developed by engineering, genetic engineering. And this is the biosynthetic pathway of cholesterol, where this provitamin D3 is converted into cholesterol and is not converted into vitamin D3. This is why you don't get vitamin in case of tomato. And therefore, in this technique, this particular enzyme, which converts provitamin D3 into cholesterol, this was knocked out so that provitamin D3 could be assembled, accumulate, and then converted by sunlight UV into vitamin D3. So this is very, very important recent investigation where tomatoes with vitamin D3 could be developed and grown. I don't know whether this has gone commercial already, but this is an important paper published in 2022. Here, see, this is the vitamin D, Science May 23, 2022. The tomatoes with rich vitamin, and there are two groups which discovered this particular, developed this technique. One led by Norwich, another led by South Korea. But there are limitations with gene editing, and the limitations are Cas9 creates double strand breaks, which is a limitation. This is a limitation because these double strand breaks lead to non-homologous end joining. You see, there are two different methods which will be followed for repair later on after Cas9 is used: non-homologous end joining and homologous dependent recombination. And this NHAG, which is not really desirable, competes with double homologous dependent recombination, and it gives undesirable results, and therefore you have a serious problem with the technique. Then target window is too long, therefore you get offsites. Indels and offsite editing is very common, and then restrictions due to PAN, proto-spacer adjust motif, must be present 20 base pairs downstream, and only then you can get the editing done. The result is that the frequency of gene editing is less than 5%. So this is shown, the same thing is shown here. Then in 2016 comes this base editing, which I am very fond of talking about. I also published a paper in Current Science on this, because you see, David Liu at Harvard developed this technique in 2016, and Nature described this man as one of the 10 most important persons in science in the year 2017, which I read, and therefore I started reading about this base editing and then published a review, and also delivered a lecture at Harvard, which Ashok attended. You see, this involves deamination of cytidine into uridine and adenosine into inosine, so that CG can be converted into AT and AT can be converted into CG. And other possible alterations in base editing were also later on developed. But one of the most important points which I want to make here is that cytidine aminase was available in plants, in DNA, but adenine deaminase was not available. And therefore, adenine deaminase had to be synthesized artificially. And this really took tremendous effort in David Lee lab. And these two ladies, which were postdoc, both of them were postdoc in Dewey's lab. One of them was the first author in 2016 paper. The other is the first author in the second paper, 2017, where she developed this adenine deaminase from RNA deaminase. RNA deaminase was available, but DNA deaminase was not available. As a result of this, RNA deaminase had to be used and converted into DNA adenine deaminase. And when you read this paper, you see the kind of effort which had undergone into developing this artificial adenine deaminase, which was then subsequently utilized for gene base editing. Then another one, and this technique was immediately followed in China. Large number of papers in 2018 and 19 were published from China, where they used this particular technique for a variety of purposes. And you can see in this particular case that the cytidine deaminase domain was used, and then this nutritional improvement could be done, herbicide resistance could be obtained, and large number of different traits could be improved through gene editing in case of plants, particularly in China. But in India also some work now is being done, but not at a large scale as has been done in China. Maize was developed, gene for sulfonyl herbicide resistance was developed, so herbicide resistant maize could be produced. Then prime editing is another important technique which was developed in 2019 because there were limitations with DNA base editing. Therefore, that technique was substituted because in those techniques of cytidine base editing and adenine base editing, you use double strand breaks. And double strand breaks in case of base editing is a limitation because it gives off types. It gives off types and large window. So in this particular case, they use reverse transcriptase protein associated with this particular RNA molecule pack and the Cas9. This complex is utilized. And in this particular case, you can see that no double strand breaks occur. There is a nick here with the help of Cas9, which takes place. And then this nick starts copying this reverse transcriptase RNA. So there is no double strand breaks involved. It is the reverse transcriptase which helps in copying the change which we have introduced in the form of this RNA. The desired alteration is introduced in the form of this RNA molecule, which is copied with the help of reverse transcriptase. And therefore, this prime editing during the last 2-3 years, discovered in 2019, but 2020-21, it was demonstrated to be successful in many other cases. Then synthetic genomics, just one slide. The most important, you must be familiar with the number of microbial genomes which have been synthesized, but the most important for us is the synthesis of yeast chromosomes. All yeast chromosomes, not all of them have been synthesized. 6 of the 16 chromosomes have already been synthesized. And then another effort was made where all the chromosomes were joined together in the form of genome sequence. And in 1 or 2 molecules, the whole chromosomes of yeast could be synthesized. Then these are the 4 exciting areas which I mentioned in the beginning, which I like to talk about very briefly. For those students who would like to really know some of the recent areas, one is the 3D chromatin and spatial genomics. This is one particular topic for which I am writing an article for Trends in Plant Science. Then Earth Biogenome Project, which is mentioned in the title. Then Bioinformatics and Protein Folding, which was declared as science breakthrough of the year 2021. And then synthetic gene drive, which became very important during the last few months. Very briefly, going through these, This is the technique which was developed in 2002, Decker. The paper was published in Science. And you can see one of the most important reasons why you should study chromatin organization is that there is protein folding, particularly because the sequences situated far away will fold and try to regulate the sequences located far away. So this folding of the chromatin is necessary for regulation of gene sequences. And one would like to understand the relationship between the gene and its regulator sequences, which is situated far away from this particular sequence. And therefore cross-linking was done, and cross-linking, after cross-linking, some labeling was done, and then following, then ultimately you have a sequence here, which is a synthesis of sequences which are located far away in the genome, but are located close to each other for the purpose of sequencing. So you are really deriving molecules which have sequences which are generally located far away, but for the purpose of sequencing, you are bringing them together, and you know them. These sequences, regulatory sequences. This is the genesis of this development of these techniques of three-dimensional chromatin. You see, as a result of these techniques, a number of them were developed actually. If you go back, you can see here that this 3C technique, chromosome conformation capture, was developed in 2002, followed by sequences which we call 4C, 5C, 6C, 7C, and then ultimately in 2009, high C was developed, which was utilized for the determination of a three-dimensional structure of chromatin. And as a result, you have this situation. In the nucleus, you have chromosome territories. Chromosomes are not randomly distributed, but they are distributed in chromosome territories requiring according to function. And then chromosome territories consist of compartments, two types of compartment, A and B. A is active compartment, euchromatin. B is silent compartment, heterochromatin. And then these compartments in their turn consist of topologically associated domains which physically interact. These are the TAD sequences. Then these are having loops here, and then loops. So you start with DNA, then nucleosomes, then loops, then you have TAD sequences, then you have compartments, and then you have chromosome territories. This is described as hierarchical model of chromatin organization within the nucleus. In contrast to this model, there is another model which describes this organization by phase separation. You can see here, this is compact, this is hierarchical model, and this is a model which they call phase separation, and they may act together. They may not be alternative models, but sometimes phase separation takes place, sometimes 30 nanometer fiber, condensed fibers are utilized. This is the situation. You see, you have bridging factor, which will bring about this polymer phase separation, and then you have another technique which will give rise to liquid phase separation. So these are contrasting models for phase separation, which say that chromatin organization is not really chromosome territories and AB compartments and TADs and so on, but they can also occur. Chromatin may also be occurring in these two forms: polymer which separate from each other, and liquid which they separate from each other. Then more recently, during the last 2 years, techniques have been developed where the role of proteins and RNA in 3D organization has been characterized and demonstrated. So there are a large number of proteins and RNAs which determine the 3D organization of the chromatin, and therefore in order to find out that what DNA and what proteins, RNA proteins are involved, two techniques were developed. One was this sprite technique, split pool recognition of interaction by tag extinction. And then there is another one, RD sprite, RNA, DNA. They have been developed at Gutman's lab and in University of California, Davis. And several important papers have been published during the last 2-3 years. You can say the three-dimensional organization of the chromatin also depends on nuclear bodies, as shown in particular case here. These nuclear bodies include Kajal bodies, speckles, and nuclear membrane. And the chromatin organization depends upon these nuclear bodies also, which facilitate the 3D organization and their function. Then Earth Biogenome, very briefly, I will tell about it. The project was launched in November 1st, 2018. The pilot phase 2018 to 20. Then 20 to 23, there is phase 1. And then phase 2, 24 to 27. And the project is targeted to be completed by 2013, where 1.8 million species, which are characterized and named so far, all of them have to be sequenced, at least one sample of each. And this is the organization of this Earth Biogenome project. There is a coordinating council. Then there are a number of subcommittees which will look after this particular work. Then next second is there are the different projects, networks, which are currently operating. And this is why also described as network of networks. Large number of networks are there, and then they are again forming a network, which is the Earth Biogenome project. And these, the outermost circle shows the countries which are involved in this particular project. These are the components of Earth Biogenome project: sample collection, sequencing, assembly, physical maps, then synteny and chromatin states study, and then public data analysis. And these are some of the outcomes of this Earth Biogenome project. And the most important is that this should ultimately help in conservation of genetic resources, because you will know the sequences, you can develop strategies for conservation. The last, protein folding, this has attracted my attention because this was declared as science breakthrough of the year 2021. And this is actually the development of artificial intelligence and machine learning, with the help of which number of these algorithms have been developed: AlphaFold, AlphaFold2, TR Rosetta, and Rose TTA Fold. And they say that utilizing these different algorithms, you can find out the three-dimensional structure of the protein without really conducting any crystallography or anything, from the sequence directly. And these have been tried, and the results obtained from this computerized exercise were completely like those which were determined. Thousands of proteins have been compared, whose structure was determined experimentally with the theoretical structure based on these algorithms. It means that you can really find out the structure, three-dimensional structure of the proteins with the help of computation. And this is going to be remarkable because number of our students find out new genes in wheat by simply computation, and then also determine the protein and its function. And if they can do it, and they have the algorithm, we have software available which our students are using, but none of them are using these recent ones, which are much, much sophisticated. You can see these papers: AlphaFold, AlphaFold2, Rosetta, and at least 190,000 plus new protein structures predictions were found, and they were exactly like those which were determined by X-ray crystallography. Gene drive is another last one, synthetic gene. Gene drive, you must be familiar with this particular phenomenon, which is described in textbooks also: selfish genes, which will spread without following any law of inheritance. But why I am talking about it, this old phenomenon which is already known, is synthetic gene drive have been developed recently during the last 5 years. Synthetic gene drive, you can artificially do it. For example, if there is a weed and you try to develop a technique which will kill the weeds and will not allow the weeds to grow in case of malaria, particularly in case of malaria, the Anopheles, which transmits malaria, this has been rendered mere sterile so that the population will not spread. And therefore, in many publications, they say a lot of scope of this particular phenomenon in future. This is the situation. This is Mendelian inheritance, and this is gene drive. You can see that in particular case, in this particular case, does not follow Mendelian inheritance, and the gene introduced or edited, it spreads throughout the population within a few years. So they say a lot of potential in this particular approach in future plant breeding. And these are the different benefits of gene drive. CRISPR-Cas technique has been used in order to develop the technology of gene drive artificially, and it is expected that this will be used extensively in future in order to destroy particularly the weed sites or any insects, particularly in animals. Anopheles has been demonstrated, but I think in case of agriculture also it is going to be used in future. This is in addition to gene editing or base editing or prime editing, which we have been talking about. So in summary, genetics to genomics is an exciting journey. Mendelian and Morganian genetics existed for some time, then biochemical and bacterial genetics. Then we had molecular era and gene synthesis, recombinant DNA and DNA sequencing, quantitative genetics starting from 1990 onwards, whole genome sequencing and synthesis 95 onwards, and then gene editing, base editing, and prime editing, and 3D chromatin organization, Earth Biogenome Project, synthetic gene drive, and protein folding. So it has been exciting for me. The invitation for this lecture gave me opportunity to learn more because I had to read more in order to give this presentation. I very much thank Ashok for inviting me for this lecture, which gave me an opportunity to learn more genetics. Thank you very much once again. If there are any questions, I would be
So I'm sure all of us enjoyed this lecture, telling us about the milestones in genetics. If there are a couple of questions, we can take them up. Professor Sharma, please. दीजिएगा जरा microphone.
Was a little curious about synthetic genome.
थोड़ा सा आप बताइए मुझे.
Synthetic genome, and it's at the highly repetitive DNA sequences and their role. Synthetic genome.
Synthetic genome
and the highly repetitive DNA sequences and their role in the nucleus.
In the nucleus, yes. You see, the repetitive sequences, earlier we thought that they are nonsense sequences. Junk DNA, we used to use the term junk DNA. But now we know that majority of these repetitive sequences play a very important regulatory role. This is why we are trying to study 3D organization of the chromatin, because majority of this repetitive DNA is playing some role in regulation of gene activity elsewhere. The coding genes are maybe 5% or in some cases 1%, but the remaining non-coding DNA plays regulatory role, and that is the part which is being studied extensively now through 3D organization of the chromatin and other techniques.
I just had maybe I understood wrong, but you said that in GWAS, you don't want to call it quantitative QTL. But
Yes.
But why? Because all papers mention QTL.
Yes, but I differ with them. You see, many papers describe the outcome of GWAS as QTLs. But when you find out the markers associated with the trait in GWAS, you do not know where the QTL is. In interval mapping, we know. You can locate the QTL. And this is why, although in many papers you call them QTLs everywhere, but I like to call them MTAs, and MTA should then be used in order to find out the QTL. Yes, but you have to use haplotypes. If you use haplotypes, then of course you can.
So
because in haplotypes, you know the causal. So in GWAS, if you know the causal polymorphism, yes.
Very fine. So thank you very much, Professor Gupta, for this nice lecture. That's for him. And as I understand, the genome is dynamic, so is the session. And therefore, there are two changes, as I have been told by Dr. A.K. Singh. Number one, there would not be a tea break. So we will continue. Number two, that in the beginning I announced in place of Professor Painter, there will be Professor Vishwanathan. But now I understand Dr. Prasanna has to leave, and therefore the next lecture will be by Dr. Prasanna, and Dr. Vishwanathan will be speaking in one of the next sessions. So that's the second change. And the third change is that now onwards, my co-chair, Dr. Sashane, will handle the session.
Good afternoon, everyone. Respected chair and all the teachers and colleagues and my friends. As ordered by Chairman Sir, Professor Tyagi, I'll be introducing the next speaker, Dr. B.M. Prasanna. Dr. Prasanna is known as the teacher among his students, and I am one of the students also. So he has done it from the Andhra Pradesh Agricultural University, Agricultural College, 1981 to 85. Then Indian Agricultural Research Institute, IRA, New Delhi, 1985 to 91. Then he is the director, CGIAR Research Program, Maize 2015 till date. Director Global Maize Program, CIMIT International Maize and Wheat Improvement Center, Nairobi, Kenya, 2010 to date. He was a scientist, a senior scientist, a ICR National Fellow, Principal Scientist in the Division of Genetics, ICR IRA, New Delhi. And the awards and honors, he has many. FAO Fellowship and at University of California, ISCA Dr. B.C. Dev Memorial Award 2001, ICGPB Dr. Joginder Singh Memorial Award, IRA Best Teacher Award 2004, ICR National Fellowship 2005, NASA Associateship, BP Pal Memorial Award. He is the fellow of NAS, Indian Society of Genetics and Plant Breeding. And he has the research area always, as usual, in maize, as he is heading different organization in maize. Now, under his leadership, CIMIT established a state-of-the-art maize double applied facility at Kenya. His team has been in the forefront in tackling the maize lethal necrosis epidemic in Eastern Africa, as well as fall armyworm challenge across Africa. Now, we are very much eager to listen to you, sir. Thank you.
Thanks a lot, Professor Tyagi and Shashani, for the kind introduction. It is always a great pleasure to come back to this institute, my alma mater, especially the Division of Genetics. And what I really enjoyed today is establishment of this beautiful statue of Mendel there, which remains and will remain a legacy for years to come. Well, let's see which is the next. Okay, I can't start my lecture without remembering that the beautiful Division of Genetics, which has served fantastically over so many decades, producing brilliant students, brilliant faculty members. It has a huge legacy, a very rich legacy of teachers who are outstanding in teaching genetics and plant breeding. And that legacy needs to be maintained for generations. And some of my teachers are here. You can see Professor Sarkar there, and a number of very distinguished teachers and very young faculty members. I am here, Prabhu at that time, and A.K. Singh here. So we were privileged to be working in this fantastic division. And I had the opportunity to teach 4 courses at the Division of Genetics for almost 20 years. The elements of genetics, the plant gene regulation, developmental genetics, and the innovative approaches in plant breeding, which A.K. Singh and I used to teach together. And let me also recall at this time Shanti Chandrasekharan, an outstanding teacher of genetics, who together we taught both elements of genetics as well as developmental genetics. So what more better quote can be there for a teacher than this one by Harriet Martineau, who said that what office is there which involves more responsibility, which requires more qualifications, and which ought therefore to be more honorable than that of teaching. So this is one of the most enjoyable phases one can ever have as a teacher of a great institution like IRA. Both Professor Dr. Balram as well as Dr. P.K. Gupta elucidated very beautifully the history of genetics. As Dr. Balram presented, it's almost now 4 decades since DNA-based markers have come into being, thanks to the pioneering work done by these 2 gentlemen, David Botstein and Eric Landa, who revolutionized the way in which we look at markers. I still remember in 1990s, there was still some discussion and debates in the Division of Genetics whether markers should be used in plant breeding, which markers should be used, whether RFLP markers or DNA-based markers will indeed be so impactful. And that debate has been laid to rest in the last 4 decades. No longer there could be any discussion on this. We have gone far, rather than simply creating linkage maps. As Professor P.K. Gupta pointed out, we went all the more into map-based cloning, identifying, characterizing, elucidating the functions of an array of genes that influence diverse aspects of plant development. Along with this, there have been tremendous advances in instrumentations. Diverse genotyping platforms have been evolved over the years. These genotyping platforms, you can say 1st generation, 2nd generation, and 3rd generation. The 1st generation platforms have not more than 10,000 data points per year, or per day rather. Today we have 3rd generation platforms that can provide you with more than 1 million data points per day. 1 million data points per day. In fact, genotyping a maize line is now much more cheaper than actually phenotyping that line across multiple locations. So that's how the advances in genotyping have revolutionized the way in which plant breeding is done. So we have low density genotyping platforms that enable you to do routine breeding applications, including marker-assisted forward breeding or marker-assisted backcrossing, or quality assessment and quality control at the various points in the value chain. You have medium density platforms that enable you to do genomic selection. Genomic selection need not be done with millions of markers. You can simply do it with 3,000 markers. That's what we do routinely at CIMIT in our breeding pipelines. Genetic diversity analysis as well as background selection in marker-assisted backcrossing. You go further down the line with very high density platforms, they enable you to do diversity studies much better than at any time before. So therefore, this is a tremendous advance in the last few decades, the evolution of genotyping platforms. All this had a tremendous influence on diverse aspects of genetics. Today, population genetics, quantitative genetics, the way in which we do genetic diversity analysis, the way we unravel the structure and function of genomes, accelerated breeding, all these spheres of science have been thoroughly influenced by the revolution in molecular tools and technologies. I will go very briefly into that before I go into breeding aspects. One of the phenomenal aspects of the advent of molecular tools, how they influence genetics, is the understanding about how genes drive development and evolution of crop plants. These dynamics of evolutionary processes associated with crop origins and diversification has been a major feature in the last 2 decades. And this gentleman here, John Dobley, is actually the guru of Ed Buckler in a way. And he is the one who inspired Ed Buckler to look into the evolutionary genetics of maize. John Dobley's contribution to the understanding of how maize has originated is phenomenal. And it is he who provided the teosinte hypothesis and how a wild grass like teosinte, through mutations in major genes, can lead to transformation into a modern maize plant. What a fantastic example of how human selection can drive an evolution of a phenomenal crop species that is today one of the largest grown croplands anywhere in the world, with nearly 1 billion metric tons and grown on 184 million hectares. And behind this, the contribution of the Mexican maize farmers is phenomenal. So maize is a crop plant that has been derived by man, derived by human selection. And the first breeders are undoubtedly who? Farmers. The first breeders are not the institutional breeders. The first breeders were farmers who actually led to the transformation of teosinte into maize. Then came the work by Barbara McClintock, which Professor R.B. Singh also talked about. And Barbara's work is not just about discovery of jumping genes. Her contributions to cytogenetics, her contributions to genetics were phenomenal. She was the one who discovered a number of genetic phenomena which were hitherto undiscovered, unraveled. And it was Barbara's Nobel lecture in 1983 actually, published in Science in 1984, which still remains a highly relevant paper, the significance of responses of the genomes to challenge. She was the one who visualized genomes are constantly facing external and internal stimuli. Sometimes you do tissue culture, sometimes you do wide hybridization. How do these trigger silent transposable elements into active ones? So she was phenomenally futuristic in her thinking. And transposons, although earlier classified as junk DNA or something like that, today our understanding about transposable elements has dynamically changed. We now know that transposable elements come in many different forms and shapes. They are not randomly distributed in the genome. They are sources of extensive sources of mutations and of genetic polymorphisms. They are associated with genomic rearrangements and unique chromosomal features. Actually, this intrinsic balance between transposable element expression and repression drives many of the nuclear features. Insertional mutations happen both in germline as well as in somatic tissues. And a number of key coding and non-coding RNAs are actually derived from transposable elements. So our understanding of transposon dynamics has changed phenomenally over the last 2 to 3 decades. There was a lot of talk yesterday about epigenetics, and I was internally, mentally thinking, we are creating perhaps too much hype. Epigenetics is undoubtedly important in terms of genetic regulation. There is no doubt about it. There is way beyond genomes that can influence the expression of genomes. That is very well understood now. But let's also recognize that epigenetics there is a long way to go. We still do not know, is chromosome resetting happening during sexual reproduction? Is it different in plants versus animals? Are epimutations acquired during the plant life actually are inherited to some extent even? Because remember, when gamete formation takes place, the epigenetic marks are erased. It would be a clean slate. It is a clean slate, in fact, that actually protects organisms from many of the things that can be damaging. So a key question, is it indeed possible to have heritable epi-alleles that could be used in breeding strategies? So understanding epigenetics as an influencer of gene expression is different from understanding epigenetics for the purpose of crop breeding. These two need to be differentiated. In clonally propagated crops, epigenetics could be a driver for changes and which could be used. But in most of the gamete forming and sexually reproducing croplands, we still have a long way to go. And as Professor Balram says, the jury is still out. But let us not create a epigenetic hype. Some authors point out to this tremendous epigenetic hype that is being created. And we need to be careful. So students, when we listen, let's have a balance. Let's read all papers. Let's understand what is possible, what's not possible. The Bandwagons, I know, is one of the best papers written by Rex Bernardo. The Bandwagons, I know. And there are so many bandwagons that come into science. We need to be understanding which are important to jump into, which are important to watch, and which are important to let go. And at the same time, when we go into developmental genetics, this is where molecular tools had a phenomenal influence. Organisms are nothing but clocks and maps. What are clocks? What are maps? Clocks means spatial expression. Maps means spatial expression. Clocks means temporal expression. It's the spatial and temporal expression which drives organ development. And this phenomenon of homeosis, was first discovered and elucidated by Ed Lewis in 1978, and Christian Naslin Wolhard, a Drosophila geneticist. And I was first introduced to Christian by Shanti and Dr. R.P. Sharma, and phenomenally gifted scientist Christian was, rather is. And she wrote a beautiful book called The Wisdom of Embryo. Read it. And The Wisdom of Embryo. And in that, Christian points out to how homeosis plays not only a critical role in animal development, but also in plant development. Another equally important aspect is heterochrony. Heterochrony is differential timing. So it's homeosis and heterochrony which actually leads to organ development and organisms development. So decades of research into the molecular mechanisms underpinning plant development. Today, we understand far better the genetic architecture of plant development, as well as the parallels and difference between the development of plants and animals. This guy is considered as one of the greatest developmental biologists that we have ever seen, John Goodall. John Goodall worked on xenopus, how nuclear transplantation influences expression in putting different cytoplasm. And John Goodall received Nobel Prize for this work in 2012, and his Nobel Prize lecture was titled "The Egg and the Nucleus, a Battle for the Supremacy." What is even more important is he is the first person who could demonstrate that mature cells can be reprogrammed to become pluripotent. And that sparked the stem cell hypothesis, the stem cell theory, and the research on stem cells flowed because of great developmental biologists like John Goodall. So animal and plant developmental geneticists had a constant interface to understand what we can learn from each other. And today, pluripotency and stem cells are equally important as much in plants as they are in case of animals. Engineering plant architecture is the result of all this and will be increasingly seen thanks to the pioneering work that has been done in model organisms like Arabidopsis, as well as rice, maize, pea, and tomato. So this led to identification of an array of new genes and mechanisms controlling shoot architecture, as well as understanding of how lateral branches form, control of stem elongation, thickness, angles, regulation of inflorescence development. And behind all this, we must give credit to the third person, Elliot Meidowitz at the Caltech. Elliot was the pioneer in starting the work on Arabidopsis. And his work on Arabidopsis, especially on pattern formation and flower development, are instrumental to our understanding of the role of genes in driving development of plants. So there are fantastic opportunities in terms of engineering plant architecture. We have a foundation of rational approaches to crop breeding and generation of idiotypes with different architectures and features that can improve both plants' performance and productivity. How did molecular tools influence quantitative genetics? This is a big question. Quantitative genetics, as Rex Bernardo says, over the years, quantitative genetics in plant breeding has become increasingly empirical and computational and less grounded in theory. This trend will continue as the amount and types of data available in a breeding program increase. Rex Bernardo is one of my favorite writers, fantastic writer he is. He's not only an outstanding quantitative geneticist, but you read his papers, they are really path-breaking. They propel you to think. They stimulate in you what is possible. So he, in his beautiful article, which I cite there in Heredity 2020, and he gives very innovative titles. He says, reinventing quantitative genetics for plant breeding, something old, something new, something forward, something blue. What is blue? Best linear unbiased estimators. So look at these titles. And what is old? Multiple loci plus environmental effects. They will remain the same, the Mendelian inheritance behind quantitative trait loci. The importance of identifying candidates with best genotypic value is still an old concept. It will continue. Continue finding major Qtls, Dr. P.K. Gupta. He says, keep doing it. It has rewarded despite initial skepticism or even cynicism. Qtls have really worked. What is new? There is no need to define different types of genetic variances. Quantitative genetics has moved far beyond. No need for a reference population in a Hardy-Weinberg equilibrium. Borrowed. What is borrowed? Borrowed from animal genetics, animal quantitative genetics, is focus on reliability and least significant difference in your breeding programs. Simulation approach to design a breeding program. That's what plant geneticists are now learning, plant breeders are now learning, thanks to the advances in animal breeding. Whether it is sheep or pigs or poultry, animal genetics is the one which gave the concept of genome-wide association state. The non-populations. And blue, what is blue? You follow mixed modal analysis using best linear unbiased estimators for fixed effects and best linear unbiased predictors for random effects. How many of you have read this paper? Please raise your hand among the students. None. That means we need to evolve as a teaching discipline. Genetics need to evolve. Faculty members need to be continuously updated. I am a continuous learner of genetics. I am a student of genetics till now. I am 57-year-old. I joined genetics division as a faculty member at the age of 25 after completing PhD. For 20 years, I taught genetics, but still what I know is extremely little. Then going into genetic diversity analysis. Maize, what a beautiful crop it is. The diversity is unparalleled. Look at the diversity of maize from Mexico. We have at our CIMIT gene bank in Mexico 28,000 landrace accessions, of which 24,000 accessions come from Mexico alone. Mexico gave maize to the world. Long before all this bloody plant breeders' rights and the restrictions on movement of germplasm, if Mexican farmers have not bred maize and the maize has not flowed across the world, today we would not be having maize. And you have all sorts of maize in Mexico. You just need to go there to enjoy it. You have the world's longest ear maize with 40 centimeters long maize. Can you see that here? This is, what is the name of this maize? Hala maize. It's pronounced as J in Spanish, J-A-L-A, Hala maize. This is the world's longest ear maize. Every year we conduct a competition in Mexico, which farmer actually has grown in the year the longest Hala maize. And there is this maize, which is the conico, which is still, we do participatory engagement with farming communities and link this conico maize to the best, what we call restaurant chains in Mexico and US, and provide the income back to the farming communities. And then there is this blue maize, which is a delicacy in Mexico and in many parts of Latin America. So Mexican maize is undoubtedly one of the richest. But India, surprisingly, for many of you young students, also harbors tremendous amount of diversity, especially in the northeastern Himalayas. As a national fellow of ICIR, I had the pleasure of spending almost 1 month going around different districts of Sikkim, traveling from the lowest to the highest elevation, collecting the maize land races. And maize, Sikkim primitive, this is called Sikkim primitive with the lack of apical dominance. Each year, there is a year, at each node there is a year. 6 to 9 years on a single plant, complete lack of apical dominance. And the Sikkim primitive is because of Teosinte branch 1. And yesterday Mahapatra was talking about TP1 and its usage in cereals. So TP1 came from crop plants, crop accessions like Sikkim primitives. And there is all sorts of maize, Seti Makai, Rathi Makai. Putli Makai is nothing but McClintock's transposable elements expressed in the pericarp anthocyanin. And there is something called Gadbade Makai, means something Gadbad has happened. And white is mixed with purple and they call it Gadbade Makai. And so you go to Sikkim, every house actually hangs this maize. So northeastern Himalayas. So that's why I was triggered to put this quote by Jules Henri Poincare. The scientist does not study nature because it is useful. He or she studies it because he delights in it. And he or she delights in it because it is beautiful. If nature were not beautiful, it would not be worth knowing. And if nature were not worth knowing, life would not be worth living. What a beautiful quote. When Dr. Balram talked about science and nature at the end, I was remembering, oh, that great man's wavelength matches slightly with mine. So take inspiration from the nature. Go ahead, visit. Identify those accessions which are still grown by farming community, explore them. There is nothing better than that. And not just enjoying the beauty, we actually tracked the way in which global diffusion of maize happened through a Generation Challenge Program project when I was still at IRA. And this is a landmark paper with 799 land races from all over the world. You look at each red dot, it shows where this land race came from. Right from Americas to the Europe, to the Africa, to Asia, including India and China. I don't think this will be possible in the present world. And we tracked how maize has, through molecular marker analysis, how maize migration could have happened from the origin to the diverse parts of the world. So we published a paper called Out of America, tracing the genetic footprints of the global diffusion of maize. Using marker analysis, this is not possible simply by doing morphological analysis or just giving some wild thoughts about how maize got introduced into India. You can do this empirically You can prove it. And today, we have completed 10 years of work on Seeds of Discovery, a project funded by the Mexican government. This is a unique project characterizing what these global accessions of maize in our gene bank actually have in terms of unique alleles. So we created what we call maize molecular atlas by looking into the not only phenotypic and passport data, but also molecular data, and provides data, tools, resources to allow germplasm bank users, including breeders, researchers, extension agents, to identify the diversity of likely value for specific needs from the tens of thousands of accessions that the gene bank has. This kind of work needs to be done in almost all the national gene banks, as well as international gene banks. That's how you unravel. These gene banks should not be just museums where some visitors come and you take them. These gene banks need to be interfaced with the breeding community. And accelerating breeding. I remember my guru, Professor Kumud Sarkar. What a fantastic geneticist he was. I learned my passion. My passion for maize came through this great man. He doesn't come to any more scientific symposia. He enjoys watching his cricket matches, no problem. But he is fantastic. He used to teach genetics in a different style, which is not always understandable to people. But when you go deep into what he teaches, you enjoy it. And this man was second PhD student of Edco at University of Missouri, Columbia. And Edco was the one who found stock 6, which is the mother of all haploid inducers anywhere in the world. And it was Sarkar who probed the genetics of maternal haploid induction in maize. Pioneering work, pioneering work. Sharad Chase, that man in the third picture, actually took haploidy to a practical application form. He worked for DeKalb for a long time. And he's not so much interested in genetics. He's interested in how to use haploidy to get better double haploid lines. And till his death when he was 96, he died. Can you believe he was in constant touch with me till the time he died? Trying to understand what CIMIT is doing in haploidy. What are you doing, Prasanna? What new developments are happening? And it was one of my biggest regrets. I could not meet him personally, but that's the kind of passion people have for their subject. Sharad Chase. So friends, young friends, why am I talking about all this? We actually live on the shoulders of giants. Let's not forget them. In any branch of science, we actually live on the shoulders of giants, not just sit. And it's their contribution that propelled me into developing these temperate haploid inducers into tropicalized haploid inducers when I joined CIMIT. In 2012, we came up with the first generation tropicalized haploid inducers. In 2017, we took it to the next level, 10 to 13% haploid induction rate, one of the best inducers you can ever see. And many public sector as well as private sector institutions, today, 30 public sector institutions, 32 seed companies, including the large multinationals, have access to these materials. That's the power of germplasm. That's the power of genetics. And these three double haploid centralized farms facilities, one in Kenya, one in Mexico, and the third one very recently established at Kunigal, 70 kilometers from Bangalore, is a state-of-the-art double haploid facility. I had to struggle for 6 years to establish this facility. 6 years. But finally it's done. I am very happy. And this will revolutionize the way in which we do breeding in South Asia. And will enable us, today 80% of all lines we use in CIMIT's maize breeding pipelines are DH lines. They are not inbred lines derived through multiple cycles of inbreeding. They enable us to do marker-assisted selection, genomic selection, and increase genetic gains much better than ever before. DH has revolutionized maize breeding in large multinationals. You need to do in countries like India. Whatever said and done in terms of molecular tools and genomics, phenotyping is still the king. Hai na, Dr. P.P.K. Gupta, right? Phenotyping is still the king. Without good phenotyping, you will never be able to reliably identify markers for some of the traits. And you still need to validate your final performance of your germplasm. You need to breed for farming communities. You are not just publishing in Nature or Science or in Tag. The purpose of our organizations is to develop impactful products that reach the farming communities. And therefore, the capacity to unravel genetic differences amongst your entries through reliable, robust phenotyping protocols, no matter what, my dear young students, you need to learn that. And if you go to our drought screening site in Kibako or in Hyderabad, you see a fantastic difference. You see this drought susceptible hybrid from a drought tolerant hybrid through managed stress phenotyping. That's the strength of a breeding institution. And come out with protocols that can be used not only by yourself, but by your partners across the world. And improvements in phenotyping are happening all over the world, high throughput phenotyping, whether it is canopy senescence or canopy cover, or diseases like tar spot complex, or ear phenotyping using a simple digital camera and then linking it with the program so that you can capture the data without shelling each and every year or weighing each and every year. In our on-farm trials as well as on-station trials, we use ear phenotyping using digital imaging and tremendously reduce the cost of manual phenotyping. And through this, there is an opportunity to reduce the cost of your breeding programs by almost 20 to 25%. High throughput field-based phenotyping. Markers. Every institution needs, every breeding program needs to have a robust marker trait pipeline that goes right from discovery to validation in independent populations, in breeding populations, technical validation of genotyping assays, and finally deployment. If the trait is amenable, and if there are genomic regions which have large contribution to phenotypic variants, you will be able to identify breeder-ready markers. We have a very robust pipeline here with a clear stage gate advancement process on what data I move, a pipeline from discovery to validation or validation to deployment. You need to have a clear stage gate advancement process. Several crop breeding programs worldwide, including in India, have released an array of varieties using MABC. But very few are routinely implementing marker-assisted forward breeding. Means actually there is no difference. There is no special purpose for markers to be used. Markers are integral part of your breeding pipelines. How many of us actually use this routinely in our breeding pipelines? Not just MABC, not many. And markers also provide you with an opportunity to have rapid response to emerging threats, especially biotic threats. In the last 10 years, there have been 6 major epidemics in Africa, causing $27 billion crop losses. Can you believe it? $27 billion. 6 epidemics. So programs have to dynamically respond to the emerging threats, whether it is climate change, drought, heat, waterlogging, salinity, or whether it is diseases and insect pests. This is an example, maize lethal necrosis, a disease that comes through combination of 2 viruses. It came from Americas. Sorry, not from America, actually from China, through specialty corn introduced into Kenya. And there is a deadly virus called maize chlorotic mottle virus. Sugarcane mosaic virus is already prevalent there. So this formed a combination and suddenly started killing maize plants. Then I went in 2011 to the farmers' fields after this sudden breakout happened. Farmers are talking about it as AIDS of maize plants. This is like an AIDS of maize plant. They had no clue what is happening. Within 6 months, we had to do extensive studies, send the samples to Ferra in UK, and identified that this is not a bacterial or a fungal disease, but this is actually maize lethal necrosis, which causes 80% to 100% devastation. 2011. By 2015, we identified markers. 4 years. And by 2018, we converted 52 elite drought tolerant but MLN susceptible cement lines that are parents of commercial hybrids in Africa into resistant versions. 2011 to 18, 7 years. Is it possible with conventional breeding? Where is the debate? That's the power of molecular tools. And now we have a cohort of elite maize hybrids with MLN resistance and farmer preferred traits. And this also led to another most important thing. There is a major recessive QTL on chromosome 6, which has a huge contribution to phenotypic variants and works across genetic backgrounds and comes actually from Kasaragod University. Dr. R.B. Singh will be very happy to know that. That line comes KS23, Kasaragod University, Thailand. And we identified that QTL, fine mapped it, and went to the genomic region and identified what actually is the genomic region. What is the gene actually contributing to resistance? Today with Corteva, we have a genome editing project, the first of its kind in maize in Africa on MLN resistance. And very soon we'll come out with genome edited MLN resistant versions. That's how quickly, within the matter of 10 years, you could prove from go from an era where we did not know anything about a disease in Africa to actually go into genome editing. And most importantly, as Dr. P.K. Gupta pointed out, there are several complex traits for which you will not be able to get breeder-ready markers. Then what do you do? How do you use them? How do you find genomic regions? So you don't need to do it. You don't need to find genomic regions. You can simply go for genomic prediction using genomic estimated breeding values, which Dr. Gupta has very beautifully highlighted. This is actually genomic selection today is an integral part of all CIMMYT's maize breeding pipelines, whether it is in sub-Saharan Africa, Asia, and Latin America. So none of our elite varieties come without some form or other of molecular marker assisted breeding. Simple. There is nothing like conventional and this thing. Either we do on double haploid lines, not either. We invariably do. We select for MLN resistance and a maize streak virus resistance. Early generation, screening large number of double haploid lines, discarding those lines which have unfavorable haplotypes and selecting only those which have MLN resistant and MSV resistant haplotypes. And go forward in the breeding stages, we use genomic selection as an integral part of our breeding process. That's how powerful genomic selection has become in maize breeding, not just in theory anymore. Continuous improvement. GS has multiple applications. You can do population improvement, selecting parents for next cycle based on GEDs. You can reduce the breeding cycle time. You can do sparse phenotyping. Not every line or a hybrid need to be tested in all locations. You do what we call genomic relationship matrix. And using that genomic relationship matrix, you can cut down on the cost of your phenotyping or germplasm testing. So 30 to 40% saving in time and cost with twice increase in genetic gains. That's the power. Haplotype based breeding. Nowadays, we are now moving away from single markers to actually haplotypes. In future, although we have not yet done even in maize breeding programs at CIMMYT or in wheat, this will be the order. This is going to be the future, where discovery of beneficial haplotypes for complex traits in cropland races. Haplotype structure in commercial maize breeding programs in relation to the key founder lines. Functional haplotype GWAS is a new variant of genome-wide association study for identifying superior haplotypes for a trait of interest. And actually, instead of using markers, you use haplotypes in genomic selection to improve the accuracy and prediction ability. These are not just theory, there are excellent papers already coming out in different crops. Institutions need to be very agile. We need to do pilot studies, see the benefit of it, and move fast. There is no need for somebody else to come and tell us what needs to be done and what need not be done. And most importantly, every crop breeding program, whether it is CIMMYT or national programs, we need to dynamically monitor genetic trends. And I know for sure 90% of crop breeding programs in many of the countries, they don't analyze genetic trends. They don't routinely monitor genetic gains in your breeding programs. Am I right or wrong? Silence, it means acceptance. So that has to change. This is a study that we have done and is going to be published in Nature Scientific Reports very soon. It's the first global study to estimate genetic trends across CIMMYT's tropical maize breeding pipelines in Eastern and Southern Africa, South Asia, Latin America over one decade, 2010 to 2020, using a vast amount of data. 4,152 advanced breeding trials conducted at 1,331 locations in 28 countries globally. Where can you get that big data? That's the power of international breeding network. And the gains are very impressive, 1.5 to 2% in stress-prone environments of the rain-fed tropics is not a joke. Even Don Buwick could not get such high genetic gains. My final thing is we need to move from varietal releases to varietal adoption and impacts. India is fantastic in releasing varieties. Thousands of varieties, thousands and thousands of varieties. How many actually see the light of the day? How many of them are actually causing impacts in the farmer's field? I know there are fantastic examples from the rice breeding unit here, and in pearl millet, and in wheat, but not many examples you can see. Still, we release a lot of varieties. And this needs to change. This mindset needs to change. So when I talk about climate resilient maize, I just summarized in one slide. This is what we could achieve in last 10 years. From a stage when we had 3.5 million hectares in 2016, and when I joined CIMMYT, it was 0.5 million hectares in 2010. Today, we have 7.2 million hectares of drought-tolerant maize in sub-Saharan Africa. That is all through extensive public-private partnerships. I don't claim credit completely for this. We developed germplasm, but we formed partnerships with private sector. Over 100 seed companies in Africa alone partner with CIMMYT in scaling up with their own money and in deploying this climate resilient maize. Not many examples you can see anywhere in the world about climate resilient genetic adoption in stress-prone environments in the developing world. And there are plenty of papers published on the impacts of this. I'm not talking about adoption. I'm now talking about socio-economic impacts. How drought-tolerant maize is not only a productivity enhancing, but welfare improving innovation. With increasing yield by 15% on average, reducing the probability of crop failure by 30% when Africa gets hit every 2 to 2.5 years with severe drought stress, and extra income of $240 per hectare or more than 9 months of food for a farmer and his family at no additional cost. This is remarkable considering the fact that millions of people still live on less than $2 per day. Less than $2 per day. And for them to have 9 months of food at no additional cost is something that I feel extremely proud of. And this impact is not just limited to Africa. Today we have in Asia, for the first time, Nepal is producing drought plus heat tolerant maize. And so is India. From a stage where we had zero adoption before 2018, today we have reached 845 metric tons, and next in 2023 we will be reaching 2,000 metric tons. This is again seed companies. 27 small and medium enterprise seed companies. Very proud of partnership with Vijendra Pal, who is sitting there. Bioseed, raise your hand. A wonderful partner. This is how impacts are created. And this is not just stress tolerant maize. Outstanding work has been done by Firoz and my team. I am very, very proud of them. Give a big hand on nutritious and specialty maize. The work done by Firoz and team is outstanding. Wherever I go, I highlight it. That what a dedicated team can do in terms of developing nutritionally enriched and specialty maize. That's why we don't even enter into this area. We don't need to work on specialty maize and nutritious maize in India. There is a partner doing it so effectively. But we do this for Africa. 8 seed companies are marketing 12 PVA maize varieties in 4 countries and reaching almost 330,000 people. Still a lot of ground to cover with pro-vitamin A enriched maize. High zinc maize, this is, I think, very important from the nutrition perspective and from immunity perspective. High zinc maize and high zinc cereals can be bred and can be deployed. We have maize varieties with more than 33 ppm zinc, and these have been already released in Colombia, El Salvador, Guatemala, Honduras, Mexico, Nicaragua, and most recently in Nepal. We need to do it more extensively in Africa and in Asia. And in Guatemala, one of the companies had a remarkable scaling up of high zinc maize. And where there are stunted children, where there is tremendous amount of malnutrition, and where maize is a major staple food, and where you have 150 kgs per capita consumption of maize, is it not a very, very important intervention? And finally, in summary, my dear young friends, especially students, genetics has evolved, breeding has evolved, and will continue to evolve. No doubt about it. But what is most important is the recognition that we stand on the shoulders of giants, and the recognition we need to be passionate about science, no matter whether you work on maize or rice or wheat or chickpea or any crop. Be passionate, but any debate on conventional versus molecular or old versus new genetics is totally infructuous. Develop a broader perspective of genetics and plant breeding. Be well versed with the best possible tools and approaches, but take a well-informed decision on what to use and what not to use. And partnerships. One of my teachers used to say, dosti karo. Dosti karo means do friendships. Dosti karo is so important now. You cannot achieve impacts by alone. No matter how great an organization, whether it is CIMIT or ICIR, it needs partnerships. Partnerships are critical for impacts. Here are the choices in front of us. Either we continue to do the same, or we make occasional improvements, or we make continuous improvement, or we make continuous improvement with innovations. Those are the choices. And finally, if you are in science, read this book on Barbara McClintock, A Feeling for the Organism. And this is written by Evelyn Fox Keller. What is life on Barbara? It's not just about science. It's about developing a feeling for the organism. So I leave with this final quote by Peter Drucker, a management guru. No organization can depend on genius. The success is always scarce and unreliable. It is the test of an organization to make ordinary people perform better than they seem capable of, to bring out whatever strength there in its members, and to use each person's strength to help all the members perform. The purpose of an organization is to enable common men to do uncommon things. Common men to do uncommon things. When organizations evolve to do that and not depend solely on genius. Geniuses like Dr. Balram are rare. We are all ordinary people. And how ordinary people can perform better than they seem capable of is the purpose of an organization. Let's do it. Thank you so much.
We'll invite one or two questions.
Yeah, go ahead. There is a mic, then we can come to you.
Young man first.
Yeah, it was a fantastic presentation. We must compliment you for that. And then there's one simple question always lingering in my mind. The area under hybrids have gone beyond 90%. Does it correlate with the gene action? Is it that out of say 1,000 genes, more than 900 genes are showing over dominant gene action? What is ailing varietal development programs? Is it that we are not able to tap additive gene action? We're not able to come out with good varietal development programs? Why are we banking or over banking on hybrids?
Okay, very good question. Hybrid or OPV deployment is dependent upon the evolution of seed sector. In countries where the seed sector is not very well evolved and farmers do not have access to affordable seed, we still in CIMIT, we still deploy OPVs, improved OPVs, especially as synthetics, by taking the best of the lines and then chain crossing them and getting those improved OPVs in the market. But when it comes to evolved seed sectors like in India, there is absolutely no point going into the OPV era. In fact, the evolved economies, the evolved countries with strong seed sector presence, they move away from double cross hybrids to the three-way hybrids and then three-way hybrids to the single cross hybrids. Vietnam now has almost 100% hybrid seed, single cross hybrids.
Why is it like this? I'm asking.
That's what I'm saying. It is because of the seed companies sector.
My point is, out of the total number of loci which influence productivity.
Now, when you come to the genetic basis or molecular mechanisms of heterosis,
what proportion must be showing additive gene action? No, zero, absolutely. No. All the genes have shown, let's say, like climate change, they are switched over to over dominance.
No, but what is important is in commercial crops like maize, in cross-pollinated crops like maize, there is a distinct, you don't need to explore how much is additive gene action or how much is dominance or over dominance. It's a fact that you can achieve heterosis. I don't want to cite a study by Dr. N.K. Singh, which is still in the pipeline. But yesterday I saw some phenomenal results about what can happen in a hybrid with regard to heterosis and gene action aspects. So even without understanding fully the basis for heterosis, maize breeders have been exploiting heterosis fantastically. You will never be able to understand the complete molecular mechanisms underlying heterosis, no matter what, even after 10 years, I will guarantee you. But there is a distinct opportunity to move from the OPV era to hybrid era, and there is a clear difference. Today, the examples that I showed in Africa, 7.2 million hectares, 80% of them are hybrids. Only 20% of them are OPVs. That's because in some countries like Mozambique or in Angola, there is a very, very weak seed sector. And there is no possibility for commercial growers to enter or to produce hybrid at an affordable seed. So we can discuss this separately, Dr. Patil. There is a lot to discuss. The student there.
I just wanted to add to what Dr. Patil asked. In fact, if you look at the performance of hybrids in 30s, 40s, 50s,
Exactly.
from 1.5 ton, 1 ton per hectare, now you have got hybrid up to 4.5.
4.5 tons per hectare.
And this is all 100% additive gene action exploitation. The entire improvement in hybrid performance is the exploitation of additive gene action. On top of that, you cannot deny the phenomenon of heterosis. It is there, and that is what is reflected in the performance of hybrid.
Yeah, we don't have to go into the basis of heterosis, but as you said correctly, evolution into hybrid requires, especially single cross hybrids, requires very strongly performing female parent with high seed production potential, and that's what we are achieving now.
I just had one not really question. But, as you mentioned that at IRI, we have done excellent work on developing biofortified maize hybrids. More than 10 hybrids have been released with, quality protein maize, provitamin A, and now many more traits coming. One of the major constraints we are finding how to scale them up. Industry is not ready to take. The seed industry is not coming forward. Even if the hybrids are at par in yield, they are not coming forward. When we approach poultry industry, they say synthetic fortification of feed is cheaper and doable. Why should we use biofortified? So how do we really create a kind of situation where we are able to push? One way is that if the government makes it mandatory, that if a hybrid is not biofortified, it will not be released. But so
that is too drastic. That would be too drastic.
So that's one challenge coming because so much of work is happening, but to make impact on the society, it has to reach the people.
Exactly. I think scaling up for this, Ashok, we do need innovative licensing models. I think that's where ICIR perhaps may have to come up with even geographically limited exclusive licensing models. Non-exclusive licensivity, we have moved away in Africa even. We do a lot of geographically limited exclusive licensing so that the seed company actually feels ownership of the product, invest their money. If I give it to 10 companies, if 2 of these companies actually not maintain quality of the hybrid, other 8 will suffer if it is marketed under the same name. But if you give on a geographically limited exclusive licensing models, then we can make a much better impact. That's my personal opinion. But I know ICR must have thought about it a lot.
Sir, the student first. So we student first. And we can, this is the last question. We can discuss during the time of lunch. Because this is going to the...
Good afternoon, sir. Thank you so much for this fruitful session, which you have given to us. Sir, as you discussed in your lecture about junk DNA. So, sir, I'm very curious to know about, like, in transposable elements at the time of copy and paste mechanism, if in case their mutation will occur, then what will be happen? Like, so it can be possible to lay the foundation of new traits in a positive manner, which can be like heritable to the next generations.
Okay, I need to now go back to my teaching days of plant gene regulation. But to keep the answer very short, transposons do have the propensity to cause what we call footprints, transposon footprints. So they can repeatedly visit a particular location and cause changes when they are active. So, and then this can drive evolution of new genes. There are a number of documented cases in maize, how a transcriptional activator has become a transcriptional repressor because of transposon-induced mutations. So it is a commonly occurring phenomenon, but at the same time, as I said, there are two things that happen. One in the somatic lineage, another is the germline lineage. What is happening in the germline lineage actually gets transmitted. What is happening in the somatic lineage during the lifetime, I may be having innumerable mutations. Not all of them are transmittable. Okay, so heritability depends upon where the mutation is happening, not because just a mutation is happening.
Thank you so much, sir.
Thank you, Dr. Prasanna.
Dr. Tomar is the last question. Okay, fine.
This is the last.
I am asking actually a question from farmer's point of view. You said CIMMYT has been releasing drought tolerant varieties for tropical nations. Recently in India, area under maize is growing tremendously.
Yeah.
And under conditions of north, a 110 days crop requires 12 to 15 irrigation.
Yes.
Now this causes disbalance between nature and the plants.
Absolutely.
What is it?
This is an absolutely correct one. And it's not only the Kharif season where we are applying excessive irrigation. You will be surprised in the spring maize in Punjab region, farmers irrigate almost 20 times a maize crop. Subsidized water, subsidized electricity, but causing tremendous harm to the soil water status. This is where we require water use efficient maize. The drought tolerant maize or heat tolerant maize that we are talking, which Vijendrapal knows, is not just a genotype that tolerates heat stress, but it also has an intrinsic water use efficiency. Dr. Zaidi, my colleague, conducted an experiment to show that you can reduce the number of irrigations from 20 to not more than 5 or 6 using drought tolerant maize. So that is water saving potential of this variety because of there are two mechanisms: water use efficiency and radiation use efficiency. Both these contribute to drought tolerance.
Maruti,
Dr. R.B. Singh is the only one who affectionately calls me Maruti. So I feel very happy to hear that only my parents and Dr. R.B. Singh.
Well, I must say it was a wonderful presentation, wonderful as ever. But let me tell you, I'm not a maize breeder. However, a little bit that I understand. If you look at the history of maize improvement and breeding and so on, we were misguided earlier, and thanks God that we came back to the single cross hybrids, and we all concentrate today on single cross hybrids.
Yeah.
This is number one. Number two, that I would like to tell you that the understanding of the additive gene effects and additive into additive interactions also, which could positively add to the performance through genetic gains, should be also looked into and mapped up, and because they are fixable. And these fixable genes, additive and additive types per se, should be seen as our friend Dr. Vasav mentions about.
Inbred lines.
Yes.
In deriving.
Inbred lines
exactly.
should be strengthened. And along with that, there should be a very strong program also on what we call the heterotic group development.
Exactly.
And this needs to be emphasized, heterotic group development. And from those heterotic group developments, we get the best of the inbreds up to 3 to 4 tons, 5 tons, and then their hybrids should have 12 to 15 tons per hectare. That approach will be more practical, and we'll follow that. Hopefully, we will endorse it and we'll follow that in India.
Thank you, sir. I think that is the way to look for. Dr. Vasal also talks about inbred hybrid approach.
Exactly.
Inbred approach towards, as A.K. Singh said,
yes,
using the additive and interaction effects to
Right.
derive elite inbred lines with better performance, and then use heterosis through the heterotic grouping
Yes,
to maximize the benefits of heterosis.
exactly. To maximize
So
the-
that is where Indian breeding programs suffered
Yes.
due to lack of distinct heterotic groups.
We have to concentrate on
We
this.
have to
So the
concentrate on
recommendation
that.
should be, we must concentrate on this
Absolutely.
very strong.
Absolutely.
Final question.
Yes.
There, as you know, probably there is a publication which you might have come across. It is called the Epomictic efforts.
Yeah.
Epomictic efforts, how much do you think is the feasibility and practicality and the possibility of the private sector to really accept and work together for getting an Epomictic F1 hybrid in maize?
Yes, I had an opportunity to interact with some of the best scientists working on epomixes, including Yuri Gross-Nicholas, and before that, Javier, at CIMMYT when they were working, as well as in Switzerland. The conclusion is that in some crops, epomixis is exploitable. In some crops like maize, where endosperm formation, it's not just endosperm formation, but endosperm embryo, that balance, it is still a long way to go. There is a lot of effort that has gone into epomictic maize, but till date, we understood better how epomixis happens. But to be practically used in breeding programs, especially in seed propagated crops like maize, it is still not a reality. So therefore, we should never rule something out completely. I would be very hesitant to do that. But as of today, epomixis is in maize is a distant reality, I would say.
Thank you, Dr. Prasanna. Thank you very much. Respected Chairman, sir, I am grateful to you to give me the opportunity to summarize. Also, I am grateful to Dr. A.K. Singh to give me this opportunity to be here and speak something. I have written down around 50 pages of all the lectures. That's why I was thinking that I should get some time, at least 1 hour to speak also. To summarize all the lectures. But it is very difficult for me to tell and summarize the lectures because all the three speakers are illustrious, and in no way I have no qualification to summarize their lectures. But still then, let me tell, Professor Balram took us to a journey of Darwin's evolution to Mendel's law. Then he started with Frederick Miescher, Griffith, and all the experiments of DNA, the genetic material by Avery, then coming to Watson and Crick, and then further going and telling the chemical nature of the DNA. That's the chemistry part of it. So chemistry always link everything together, and without chemistry, there will be no biology itself. Ultimately, he went to the nature, and nature means science is the nature. The same thing also told by Dr. Gupta. Then he took us a journey after the DNA discovery, how the science progressed. Then he talked about different mutations. He talked about different markers and how the markers are being utilized for crop improvement, and ultimately the genes and genomes and the post-genomic era, and how different science is going in different directions through structures of nuclear, all these things, how this can be utilized for crop improvement. Finally, we listened from Dr. Prasanna about again the markers and how he is utilizing these markers for the crop improvement, also the double haploid induction and double haploids, and how he is improving the maize system. So, and his message was to have collaborations, friendships. So that alone we can grow, we cannot grow, but with collaboration we can grow further. So these are the messages today we got. And for the, it was really a journey of genetics and molecular biology, you can say, coming together and giving us sufficient knowledge to grow and improve the crop plants. So really, I sincerely thank all the three illustrious speakers, and I have no qualification also to talk about them, but still then, as ordered by Chairman Sir, so I am speaking something here about the lecture. Thank you very much. Thank you.
Now I would like to request Dr. A.K. Singh, Director IRI, to facilitate chairman of the session with memento, which is a Mendel bust. Next is for our co-chair. Also for the speakers. Sir, for Dr. P.K. Gupta. Sir, you can take. I would like to request Dr. Balaram sir to come on the dais. Nonsense. Prasanna sir, please come on the dais. Dr. Prasanna, please. At the end, it's a customary to give vote of thanks. So, good afternoon to one and all. I am Dr. Gyan Mishra, Principal Scientist at Division of Genetics, IRI. It has been such an honor to be part of this wonderful event. On behalf of Genetics Club and our own behalf, I deem it a great honor to propose vote of thanks to all who have helped us in making this session such a resounding success. First of all, I would like to propose our hearty thanks to the chairman, Dr. Akhilesh Tyagi sir, and the co-chair, Dr. A.K. Sasani sir, for conducting today's session in such a wonderful manner. I would also like to thank Dr. P. Balram sir, Dr. P.K. Gupta sir, and Dr. B.M. Prasanna sir for their excellent presentations and making this symposium, and particularly this session, a very interesting and meaningful one. Thank you, sir, for your stability and thought-provoking addresses. I am sure that all of us, be it a student or a scientist, are equally benefited from your presentations, which touch upon like different areas of the genetics, like role of chemistry in Mendelian genetics, especially that part of what is life, and then Mendel to Qtl and the biogenome project. So many new terminologies we always used to hear from Dr. P.K. Gupta sir's presentation. I've been hearing like this from last 24 years. Then molecular tools in genetics and reading by like unique way, style of presentation by Dr. B.M. Prasanna sir, in such a way that we all got like novel perspective of our understanding about the Mendelian genetics and in this era. Finally, I would like to thank all of you present here for making time to be with us today and helping us in making this session a grand success. Thank you one and all. Now it's time for lunch break and we'll be assembling here again at 2:30 p.m. Thank you for the next session. Thanks a lot.